My name is Victor Dorita, and I'm the president of the American Society for Microbiology. And I'd like to welcome you to this symposium on microbial sciences research in the post-COVID environment. And I do that on behalf of my colleague, Stefano Bertuzzi, the CEO of the ASM, and our 30,000 members whose interest in microbial sciences brought us all here today. And I look forward to what we're going to learn about. Now, since the start of this pandemic, the ASM has really been on the front line of what is obviously a microbial issue. We early on established a registry that provides high quality information on our website and vetted by experts. We have been at the forefront in defining what rigorous science is so that policymakers understand what we can do and what we should do. We've worked closely with our clinical microbiologists, our frontline clinical microbiologists, to establish protocols and standards for testing. We've been disseminating high impact peer-reviewed science in our journals in a very quick fashion so that the world, not just scientists, but the world who needs this information has it at their fingertips. In our virtual microbe meeting this summer, we were fortunate to have two members, prominent members of the White House Science Coronavirus Task Force, Drs. Deborah Birx and Anthony Fauci, present their perspectives on this pandemic. So we've been present all along, and we continue to be present in this pandemic as it moves forward. Our Council on Microbial Sciences had an excellent session back in March to talk about what the science was that was needed for the COVID pandemic, for us to confront it in the most rigorous fashion. So, as I said, this is fundamentally a microbial problem, and the world's largest society whose mission is to advance and promote the microbial sciences has been at the forefront. And now we look to the future, because at some point this pandemic will end. We've had great news about potential vaccine progress yesterday. So we look to the future and what the science will look like in the post-COVID era. We have a number of experts today talking about issues ranging from, again, what rigorous science should look like and how we convey that to the public and to policymakers, to what the public health infrastructure should be in a post-COVID world, and what new tools we're going to be using to study microbes and to predict future pandemics. So we look forward to all those sessions today. I want to thank Win-Win, our Academy director, for the great work that he and his team have done to put this together. I also want to thank Susie Fleischig. Dr. Susie Fleischig is the chair of the Council of Microbial Sciences and a professor at University of California, Berkeley, and Arturo Casadevall, the chair of the Department of Microbiology at Johns Hopkins University and the chair of the Academy Board of Governors. And it's the Academy and the Council of Microbial Sciences that has been leading this effort to get us all here today. So I look forward to today's program with great interest, and I know many of you do as well. And so I'd like to turn it over to Dr. Casadevall now. Thank you.
And I want to reinforce some of the things that Vic said. We are in the midst of this pandemic that is already a tremendous effect on science and the scientific community. We have seen interruptions of work. Fortunately, most work is resuming in many institutions, although in many ways at a reduced pace. You have seen redirection of research work. People who were working in other areas have changed their work to focus on COVID. We've seen the cessation of in-person meetings. This is a huge change to the way in which we used to communicate. On the other hand, we've seen the rise of the virtual space for scientific dialogue. That's where we are today. We've seen some increase in productivity in some sectors of science. For example, journals are getting a lot more papers. Question is that getting some of the work that was already done out, or is it the result of changing in how work is done? More importantly, we've seen a validation of the scientific mission. And to paraphrase some of the comments from my Canadian colleagues, science is the exit strategy. So as we meet today, the United States is experiencing over 100,000 cases per day. and with such high numbers, it means that in the days ahead, we are likely to see a great increase in mortality because a lot of these patients will get sick. So this may be the worst days of the epidemic, may be the next few months. However, we're seeing good news. As Vic pointed out, we heard about good results of the vaccine. Yesterday, we also heard about Lilly's monoclonal antibody receiving emerging use authorization. And since March, new therapies, science has delivered new therapies, steroids, remdesivir, convalescent plasma. The situation today is very different than it was in March. And in some ways, we can look at this progress with great optimism. So even though we are in the midst of this calamity, I think in this today, we're starting to process the lessons learned, even though lessons learned is probably a continuing process as people will look back at this time. COVID-19 is a plague, and those is one of the classical fourth men of the apocalypse. The other ones being war, famine, and wild beasts. But COVID-19 has changed our world and may be the greatest calamity that humanity has experienced in several generations. The closest example from an infectious disease point of view is the 1918 flu pandemic, and it had profound changes to society and science. For example, in the United States, and this is a negative, the failure of the then medical scientific establishment led to a reduced confidence in science and the rise of alternative therapies in the 1920s, something that we need to watch for and constantly emphasize how science is, in fact, the way out of this calamity. On the other hand, in many other countries, the flu pandemic catalyzed the strengthening of public health and socialized medical systems. And from a societal point of view, the 1918 calamity was a communal experience and led to differences in the way people look at disease rather than being from an individual responsibility to a communal responsibility. So I'm very optimistic in the view that we will control COVID with science and public health, and this will allow us to put society back together. In fact, what I'm very hopeful of is that a human success against COVID will provide wind in the sails to take us through what is the greatest existential crisis of the 20th century, which is climate change. In other words, if we can show that we can, as a global community, take on this virus and defeat it, perhaps that will help rechannel efforts to control climate change. I want to restate the purpose of this symposium, which is to bring influential researchers and leaders from academia, government, and the private sector to discuss lessons learned from the pandemic, explore new partnerships, and suggest actionable steps to prepare the microbial science research community for the next pandemic. The American Academy of Microbiology and its fellows, as you know, is the honorific arm of the American Society of Microbiology. Many of our fellows are engaged in the fight against COVID. And this event is a joint partnership with the Council of Microbial Sciences. Both the Council of Microbial Sciences and the American Academy of Microbiology can be thought of think tanks that provide advice to the society in general. And with that, I want to introduce Suzanne Fleisig, who is the chair of the Council of Microbial Sciences, and she will tell you further. Suzanne?
Before I begin, I'd just like to acknowledge all of the members of comms who have helped us to really put this program together and thinking about what the issues are that we should be discussing. The Academy brought this to us and invited us to partner with them. They had already put together a whole lot of ideas, and we had a lot of feedback from comms that contributed to really developing it to where it is today. So I'd really like to thank everybody for their input. I'd also like to thank the staff, and particularly Win-Win, for this massive effort of putting this all together. It's been a wonderful experience working with both the staff and also the Academy in this partnership. So just following on from Arturo's comments, what I'm really hoping is that in addition to really coming out with some concrete things that relate to this pandemic, is that I'd like us to think about the positive aspects. I know that's a funny thing to say in the midst of all this trouble, but from what I've been thinking about recently is just all of the fantastic things we've been learning from this experience. For many of us, we've found that we've not been able to continue doing what we were doing before. It's almost like we've been forced to be on sabbatical, and this has given us a chance to really revisit how we do everything, what we do. For others, other people have actually had the rug pulled out from under them, where they've just not been able to continue, and they've suffered a lot. Programs that have fallen apart, lost funding, and various other issues. But basically, we've all been really forced to sort of sit back and think about what's going on. How do we do things? And I think that even before this pandemic, we really did know that we had a lot of problems in the way we went about things, but we never really had a chance to think about it. We just kept going. There was this inertia on change. So I think that there's some real opportunities here for us to when we restart and get back to normal, to do things in a different way and a better way, taking into account all the things that we've learned. The other thing that I'd like to really have to think about is that the problem with this pandemic really relates not just to the pandemic itself and solving the pandemic, but everything that we're learning and the relevance of what's coming out of this extends to all of the microbial sciences and beyond that to science in general. And actually, we had a pre-meeting for panel 3. And one of the things that occurred to me during that panel meeting, the pre-meeting, was that the relevance actually extends beyond science itself to other institutions. And the reason I think that is that we're learning about the broad range of topics and disciplines that really relate to the threat of pandemics. So to solve this, it's not just microbial sciences, but all these other disciplines and even organisations outside of science that have had to be involved in directly combating this particular pandemic. But the other thing is that I think that the pandemic itself is teaching similar lessons across fields. So it's not just the microbial sciences that has had to stop and think about what it's doing or do things differently. It's the same for every discipline. And so, I think that what we're going to be talking about here today is relevant in a very broad context. So with that, I would like to move on and introduce our keynote speaker, who I think really exemplifies some of the things that we've been discussing. I think that Dr. Krogan, who is a professor at the Department of Cellular and Molecular Pharmacology at UCSF, what his group has been doing in collaboration with so many groups across the world really will help us to think about how bringing down the barriers between different disciplines and different groups, forgetting about who takes credit for what and just going for it, sharing tools, reaching out to other countries, and not just sort of thinking about our own little world, and what can actually be accomplished when you do that. So, as I mentioned, Dr. Krogan is at UCSF. He's part of the Quantitative Biology Initiative and also the Gladstone Institute. His research has focused on HIV, human protein complexes, but he's also the co-director of 3 cell mapping initiatives. So one of them is the Cancer Cell Mapping Initiative. Another is the Host Pathogen Map Initiative and the Psychiatric Cell Map Initiative. So you can see that his expertise extends into multiple different areas in systems biology. So these initiatives map the gene and protein networks in healthy and diseased cells, with these maps being used to better understand disease and to provide novel therapies to fight them. His group has led the work to create the SARS-CoV-2 interactome and assembled the QBI Coronavirus Research Group, which includes hundreds of scientists from around the world. His research focuses on developing and using unbiased quantitative systems approaches to study a wide variety of diseases with the ultimate goal of developing new therapeutics. He's authored over 250 papers in the fields of genetics and molecular biology, and he's given over 250 lectures and seminars around the world. He's a Searle Scholar, a Keck Distinguished Scholar, and was recently awarded the Roddenberry Prize for Biomedical Research. So with that, I will hand the podium over to Dr. Craven.
So it's a great pleasure and honor to be here to tell you about some ongoing work under the umbrella of the QCRG, as Susie said, the QBI Coronavirus Research Group. And this was initially a group of over 40 scientists here at QBI and at UCSF that have come together, bringing their diverse skill set in an integrated way to try to come up with solutions for this current pandemic that we're all dealing with. And it doesn't just comprise scientists at QBI and UCSF. As Susie said, we have collaborators around the world, both academic institutions and in industry, big pharma and smaller companies. And I'll touch on this at the end. I think this is one of the silver linings, as you've heard about in the introduction, to see all these scientists around the world coming together and working together in very unprecedented and exciting ways. And the challenge is to try to keep this infrastructure and the spirit in place as the dust settles on COVID-19. So here's a couple of papers that have recently come out from QCRG. Susie alluded to one here on the left, this SARS-CoV-2 protein-protein interaction map that we had generated in an attempt to identify drugs for drug repurposing. I'm going to talk about this for a couple of minutes. I'll allude to this paper, although I won't go into the details. We also globally looked at phosphorylation changes in infected cells. It's a similar group of scientists involved in this, and we identified the kinases that were most misregulated and identified a number of kinase inhibitors that had potent antiviral effects, many of which are now being taken to the next stage. But first, our efforts focused on trying to generate a protein-protein interaction map. And back in January, when this virus seemed to start being a problem, Dave Gordon, a postdoc in my lab, actually was the first to synthesize all of the genes individually, approximately 30 of them. And we put affinity tags on them, expressed them in human cells, lysed the cells, purified the proteins using an affinity tag. We had used 2x strep initially, analyzed the material by mass spectrometry, and then came up with a SARS-CoV-2 human protein-protein interaction map. And when we initially put this map on bioRxiv at the end of March, we just simply tweeted that we had this set of genes, these set of constructs, and we'd be happy to send them out to whoever wanted them. No MTAs, there were no lawyers around anyway, which was a good thing. No strings attached, and please feel free to distribute. And in a couple of weeks, we actually sent these plasmids out to almost 400 labs in over 40 countries. And I like to say that these plasmids went around the world a lot faster than the virus did. So we're very happy that we're able to share these reagents to help expedite research on SARS-CoV-2 and COVID-19. So here's a version of the SARS-CoV-2 virus human protein-protein interaction map that we had published a couple of months ago. There's 332 human proteins in total, connected to 26 different SARS-CoV-2 proteins. And if the node here is orange, we think it's a druggable target. And we mapped the host factors to 69 different drugs and compounds. This was led by Kevin Choquette and Brian Choquette here at UCSF. And we collaborated with Adolfo Garcia Sastre at Mount Sinai Hospital in New York and Marco Vignuzzi at the Pasteur Institute to test these drugs and compounds to see if they had any antiviral effects. Here's a number that we were looking at in more detail. There's 2 translational inhibitors. Pladepsin aplidin is one. This is a translational elongation inhibitor that came from the map. Another one here is zotatofin. This is a translational initiation protein. I'm going to come back to pladepsin in just a minute. And we're also been looking more deeply at drugs and compounds that modulate the sigma receptor 1. And this is work being led by Brian Choquette. And I just wanted to point out, a couple of weeks after we published this initial map, there had been 13 drugs from this paper, derived from this paper, that were tested in clinical trials, and actually many more since the middle of May, many of which we're following closely. But I wanted to talk a little bit more about this particular drug here. It's pladepsin or aplidin. It's actually a drug that's being used to treat multiple myeloma in Australia. It's from a company, PharmaMar. This has been approved for treatment of multiple myeloma. And in work that has not yet been published yet, from Chris White and Adolfo Garcia Sastre, in human cells expressing ACE2, they actually found that this IC90 value from pladepsin is in the picomolar range, and it's 30 times more potent than remdesivir. And you look at primary lung cells, Apladin is much more potent than remdesivir, about 10 times lower concentration here to see the same effect. And then Adolfo used this mouse model where you can actually infect the mice with SARS-CoV-2. Many of you have this now. And at 3 different timings, you're adding either Apladin or remdesivir, and a much lower concentration of Apladin, you can see that it's incredibly potent, about 2.5 logs decrease of infection. And a couple of weeks ago, PharmaMar announced positive results from a phase 1, phase 2 Apladin trial. They're now transitioning to phase 3. And interestingly, where this compound comes from is this creature here, the sea creature, and that's the compound right there. And it's actually as a structure here, not from us, but from a previous group, showing that Apladin binding to EIF1A, which was a connection that we had derived from our protein-protein interaction map. And actually, when you CRISPR a specific point mutation in EIF1A, you make the cells resistant to Apladin. So we're very excited about this particular drug, and we're hoping that you'll be hearing about it very soon as an option here for treatment for COVID-19. So the last part of my talk, I just want to highlight a more recent paper that came out a couple weeks ago, where we started to look at other viruses in the past, other coronaviruses that have reared their head since the turn of the century, including SARS-CoV-1 and MERS. Obviously, we're in the middle of the SARS-CoV-2 pandemic. Well, the goal here was to look back here at SARS-CoV-1 and MERS to help inform the next pandemic, SARS-CoV-3, because we know it's coming. So can we be better prepared for the next pandemic? And can we look at the previous coronaviruses to help us get prepared? So this was a work that was published in Science a couple of weeks ago, another highly collaborative effort involving 200 scientists from 16 different institutions in 6 different countries. And the reason we could move so fast is because we all work together. And this is this comparative host coronavirus protein interaction networks reveal pan-viral disease mechanisms. So here's an overview of the study. Just like we did for SARS-CoV-2, for SARS-CoV-1 and MERS, we cloned out each one of the genes, put affinity tags on them, expressed them again in human cells, and purified the tag proteins, and analyzed by mass spectrometry the co-purifying human factors and to generate protein-protein interaction maps. In parallel, we also expressed all the individual proteins and used microscopy to determine in which subcellular compartment each protein was localizing to. We also generated antibodies against 14 of the SARS-CoV-2 proteins, so we could look at localization in infected cells to generate these cellular localization maps. We merged this together. This informed functional analysis, structural analysis, as well as a connection to real-world clinical data analysis. So as I said, just like we did for SARS-CoV-2, we generated a protein-protein interaction map for SARS-CoV-1, for MERS. And there's a number of different ways you can compare these 3 maps. Just a very simple and powerful way is to simply cluster these protein-protein interactions, which is done over here. And what you can see is about 1/5 of the interactions are shared between all 3 viruses. About 1/4 of them you see just for SARS-1 and SARS-2, and about 1/4 or 1/5 of them you can see are specific for MERS. So we're particularly interested in these 3 groupings highlighted here on this differential interaction map. We generated this map in collaboration with Triadica in San Diego. There's a bunch of math here I'm not going to go into, but I'm telling you, we're highlighting these 3 clusters in this map, and the edges correspond to which category of cluster we're looking at. So if it's black, it means there's interactions with all 3 viruses, blue, MERS specific, and red, they're specific to SARS-1 and SARS-2. So there's a wealth of information in here, and there's a bunch of hypotheses that are being generated that we and others are testing around the world. So as I said, we're also looking at where these proteins are localizing to in the cell. And this was done in collaboration with Andrew Pettin in Sheffield in England and Robert Gross at the University of Freiburg in Germany. So we're individually transfecting in each one of these proteins from all 3 viruses and looking at which subcellular compartment they're localizing to. For about half of the SARS-CoV-2 proteins, we actually have antibodies against them. So as I said, so we could look at them in infected cells. And this was work done by Massimo Pellemerini at the University of Glasgow in Scotland. So there's a wealth of microscopy data that we've generated during the course of this study, and all this data is available for people to look at. What we tried to do was to summarize the localization data in these tables, and this is a summary of the localization data when you individually express each one of the viral proteins from the 3 viruses. And here's the compartments on the x-axis here, so Golgi apparatus, ER, cytoplasm, etc. So the more blue the box is, this is where we think the protein is localizing to. And as I said, we've also done localization in infected cells for about half of the SARS-CoV-2 proteins using endogenous antibodies. And the summary here is on the left in these boxes in red, orange, and yellow. So there's some similarity here, but there's some differences as well. So there's a wealth of data here as well that has now been released to the scientific community, which we and others are using in various ways. I just want to highlight one particular localization pattern. It's with ORF9b, an accessory protein that exists in SARS-1 and SARS-2, but not in MERS. You can see it's localizing to the mitochondria here. It's pretty much the only viral protein localizing to the mitochondria. And we also see it, because there's a red box here, using the endogenous antibody. And this is the staining pattern using the endogenous antibody in infected cells. And this is kind of a quintessential staining pattern for the mitochondria. So I'm going to come back to this in just a minute, but I just want you to remember that ORF9b localizes to the mitochondria. So in parallel, what we were also doing was trying to genetically validate the 332 SARS-CoV-2 interacting human proteins that we deduced in our original study. And we did this two ways. In collaboration with Marco and Veronica at the Pasteur Institute, they used RNAi to knock down each one of these proteins in A549 cells expressing ACE2. Then using RT-qPCR as a readout, we looked to see what effects that had on infection. In parallel, we collaborated with the company Synthego, and we worked closely with two good guys here, Jared and Kevin, and what they did was knocked out each one of these genes one at a time in CACO2 cells. We sent these cells to Chris Bassler at Georgia State University in Atlanta, and he carried out a different assay for us, looking at an antibody against one of the viral proteins, an anti-N antibody, as a readout for infection. So we had two different cell types, two different genetic perturbation strategies, two different assays in two different continents to derive a set of genes, 73 in total, which we determined had an effect on at least one of these cell types. So the red dots correspond to genes that, when knocked down using RNAi, had an effect on infection, again, in A549 cells. And then in yellow are genes that, when knocked out in CACO2 cells, had a big effect on infection. And in orange are ones that we see in both cell types. So there's some similarity, there's some differences. But in total now, we confirmed out of the 332, 73 of these genes had an effect on infection. And here's a summary of those genes, where the blue balls here, the blue nodes, correspond to those genes. And then the edges are connected to proteins from one of the three viruses, or all three of the viruses, from our protein-protein interaction map. So the red diamonds are SARS-CoV-2 proteins, the orange ones are SARS-CoV-1, and the yellow diamonds correspond to MERS proteins. So I want to zoom up on one of these connections here. It's a connection between ORF9b, which I told you previously localizes to the mitochondria. So with both SARS-CoV-2 and SARS-CoV-1 ORF9b, we find it interacting physically with TOM70. And when you delete TOM70, you have a big effect on infection with SARS-CoV-2. And consistent with its localization, ORF9b, to the mitochondria, TOM70 is also a mitochondrial protein as well. So this is very consistent. So what we've been doing is marching through a number of these interactions in the context of one of our subgroups of the QCRG, the Structural Biology Consortium, or subgroup, which is led by Oren Rosenberg and Klim Verba. Klim Verba is a QBI fellow, and they've put together 60 different researchers at QBI and UCSF, mostly students and postdocs, mostly trainees from 18 different labs. And I would argue they're working together in a very unprecedented way, marching through each one of these interactions that we're functionally validating and trying to structurally characterize them using crystallography and cryo-EM. And what they did was to generate a cryo-EM structure of TOM70 here in green, binding to ORF9b, which is in orange here. And what you can see is ORF9b is tucking itself in the active site of TOM70. So what does TOM70 do? Well, it's a chaperone for the mitochondria. It exports proteins in and out of the mitochondria. So what is ORF9b doing with it? Well, maybe it's using it to get itself in the mitochondria to wreak havoc, or maybe it's inhibiting the transport of other human proteins in a way that's beneficial for the virus. We're looking into more detail with respect to how it's having its effect. But here's some more detail on the structure. The interaction is mostly hydrophobic, with 36 of the 38 ORF9b residues interacting with TOM70. When you look more closely, very interestingly, from our phosphorylation work, which I didn't tell you about or go into details with, we find two residues on ORF9b, S50 and S53. And if these are phosphorylated, there's no way ORF9b is tucking itself up into the active site of TOM70. So we think that these phosphorylation sites are actually inhibiting the interaction between these two connections. So I just want to show you one other structure here with respect to ORF9b. So when ORF9b is by itself, it forms a dimer. So here's two molecules of ORF9b, and it has a particular configuration. It's mostly this beta sheet configuration. But when it's by itself, it has a completely different configuration when it binds to TOM70. You can see this pretty much as an alpha helices here. when it tucks itself into the active site of Tom70. To me, this is remarkable how, and very rare, how a protein can have such a different shape depending on who it's interacting with. And maybe this is more of a common feature of these coronaviral proteins, is that they can have many different shapes depending on who they're talking to and what they're doing. And this is a way for this virus and these viral proteins to have a much bigger impact during the course of infection. Maybe this is a reason why this virus is so problematic. And I love this example too, because it's a combination of proteomics and genetics and biochemistry and virology and structural biology to come up with this node, which we think is very important for infection. It involves scientists, as I told you, from around the world. And right now we're looking at the structure in collaboration with scientists like Brian Scheuchert, are making predictions about compounds and drugs that could come in and hopefully, if not inhibit this interaction, but at least inhibit the function of this hijacked Tom70. We're marching through these interactions too, using cryo-EM mostly to characterize these connections. There's a couple of other QCRG papers that recently came out. This one came out last week. This was led by Ashish Mangalik and Peter Walter, and this one from Jim Wells, and this is where they've described nanobodies essentially that inhibit binding of ACE2 to spike, which could have therapeutic value. The QCRG group structurally characterized these in both one of these papers, so we're excited about this as a potential as well. And I just want to end by talking about some of the things that were touched on by in the introduction from Arturo and Susie. So clearly this has been a huge problem, this pandemic, but in my mind, there has been this silver lining. And the silver lining is the realization of how fast we can move when scientists work together, when silos are broken down. And these are silos across different laboratories, across different institutions, and between the academic world and the pharmaceutical world. And I have to say, I have serious problems with the scientific enterprise. A huge problem here is that it's set up to reward the individual so much more than to reward a collaborative group. One person gets an award, one person gets tenure, one person gets a score on a grant. But what you can see here during this pandemic is how fast we can move when we all work together. And the question I have is, can we keep this infrastructure in place when the dust settles on COVID-19 so that we're in a better position when the next pandemic comes, and it is coming? And another question I have here is, why aren't we working on all disease like this? Why aren't we working on breast cancer and Alzheimer's and Parkinson's in this same fashion? Because we can move so much more quickly. So I think as a scientific enterprise, the goal here is to keep this type of spirit and infrastructure in place. At least that's a high priority for myself. So I acknowledged a number of people, obviously, around the world that we've been working with. This is a picture of my group that was taken a couple months before the shelter in place was implemented here in March. I just want to highlight a couple of people here. So Dave Gordon and Gwendolyn Zhang led the protein-protein interaction study that I alluded to, the initial one. Mehdi focused as a postdoc on coordinating the phosphorylation work, which I briefly talked about. And Danielle Swaney, Ruth Huttenhain, and Robin Keck, these are fantastic mass spectrometrists that all work together, that are a crucial part of our lab and are instrumental to all the work that we're doing, not just on COVID-19, but all the disease areas. So I thank them, and I thank all our collaborators, and I thank you for your attention. I'd be happy to take questions now.
Nevin, that was a terrific talk. I was struck very much by your last comment about the rewards in science, because you know, it's very interesting. The structure of science and how we operate, it's in fact doesn't get a lot of discussion. But yeah, I mean, Ferric Fang and I have been big advocates that the structure should reward fields, not individuals. And I think what you're seeing here, for example, if you gave the Nobel Prize to a field, everybody could celebrate, everybody could get their diploma and put it up in the floor. So I was very struck by that, and I hope people in the audience reflect about those comments. I have my scientific question to you has to do with, in your talk, in particular, that beautiful analysis you presented, a lot of the focus was on differences. But you know, as a clinician and physician scientist, I'm struck by the similarities. When people die of MERS, SARS, COVID, the end result is the same. It's the immune system throwing everything at the lung, wrecking the lung, and affecting. So I'm wondering whether in your data there is some hint at the common pathway more than the differences between these pieces.
Yeah, there's a number of ways to look at these maps, and it's really depending on your question. The one I highlighted was, you know, I put together SARS-1 and SARS-2 because they're more similar. MERS and compared it to MERS and said, look at these differences. But there are a number of interactions, like I highlighted, about a fifth to a quarter of all interactions you see across all three viruses. And there's a number of kinase pathways, phosphorylation pathways that are common to all three. And we've found kinase inhibitors that are that at least inhibit SARS-CoV-2. We're looking to see if they have similar effects on SARS-CoV-1 and MERS. And the translational inhibitors that we're really excited about, the aplin and plinepsin, as well as the zotadifin, which I briefly mentioned, we think they would also have potent effects against MERS and SARS-1, just like we see for SARS-CoV-2. And we're presently working with labs that work on MERS and SARS-1. There's not that many out there. Matt Freeman is one at the University of Maryland. So we're presently testing those as well. So I completely agree. So we're looking at differences. We're also looking at commonalities. And one goal here is to find a pan-pathogenic therapeutic strategy so that we're, if it works on MERS and works on SARS-1, or works on SARS-2, it should work on SARS-3, right? So that's one of the big logics there. And just to your earlier point, Arturo, about the reward system, I think what's key, and I forgot to mention this, is that what's important, if you set up a reward system that rewards collaboration, like certain disciplines like physics do, they do it a lot better than some of these other disciplines, it encourages the younger scientists to collaborate. And the younger scientists have the best ideas. You want to encourage them to collaborate. So if we can change the system and encourage younger scientists to share their ideas, then I think we'll all be a lot better off. But you got to get the younger scientists the credit. You can't have these older scientists, soaking up all the credit. They have to step back and acquiesce and let the younger scientists step up and get the spotlight and get the credit as best as possible.
Thank you. We have time for some questions. You mentioned that let other studies, Susie, the rest of it didn't come through. Does that mean that you're still giving credit to one person? So let me read it to you, Nevin. From Susie Plyce. You mentioned people that led other studies. Does that mean you're still giving credit to one person?
I mentioned the people that led the other studies.
Right. You mentioned the people that led the other studies, obviously. I guess what she's saying is, when you get credit at the end, you were very generous with your credit. Obviously, you gave it to the heads of the labs or something. And does that mean that one's still giving credit to one person?
Well, I tried to list all the authors on all the papers. I did list all the authors on all the papers. So, and I gave credit to the lab heads, but then I tried to give this credit to the ones in the labs that led the study as well. So. There's some other questions here in the Q&A, I see.
You see some? I don't see them yet. But maybe you can answer them if you can see them.
Yeah, how do you envision changing the fields broadly to move faster in some of the diseases mentioned? Yeah, and that's a great question. That's what I guess Arturo and I had been discussing, trying to change the infrastructure overall in general. I know it's a hard thing to do, but in different areas, different federal funding agencies, different kind of different awards. If you could give them more to groups, I think it would encourage collaboration. And if you could reward scientists, especially younger ones, for collaborating and not penalize them. We all sit on these study sections and people ask, well, why are there so many authors on this paper? I see that as a good thing. A lot of people see it as a negative thing. So I think just kind of changing the mindset would go a long way in helping across all these different disease areas.
So we have a science question. Does the ORF9b shape shifting require a folding partner domain from the host protein, similar to how immunoglobulin folds work within the MHC? Fascinating observation.
Yeah, that's a great question. What I will say is that there is a chaperone HSP90 that Dave Agard has been worked on. Actually, Dave Agard, a collaborator, he actually had the structure of Tom70 several years ago. That was well before COVID-19. And then we found that ORF9b connected to Tom70, and we were a lot further ahead in terms of structurally characterizing it. But there's a chaperone HSP90 that binds to Tom70 and helps it with respect to its folding. And we think maybe HSP90 is also involved in manipulating the shape of ORF9b, and maybe ORF9b is using HSP90 to manipulate itself from one shape to another, and we're looking at this in a greater detail right now. There's one question here that studies of combinations of aplidin and remdesivir.
And remdesivir, yeah.
Yeah, in this particular study that I alluded to, which is hopefully will be out soon, Chris White and Adolfo Garcia Sastra's lab did combination. It's additive. You know, combining both of those together, it's additive. It's not multiplicative, but it's not any worse. So we think that maybe a combination study could be useful. The other thing I'd say about combination studies, which is quite interesting, aplidin has been used, as I said, for multiple myeloma in the past. And there was an aplidin dexamethasone trial that was done for multiple myeloma, ironically, and it was deemed to be a safe combination. So maybe this could be a combination between aplidin and dexamethasone in terms of a combination strategy for COVID-19.
So we have 3 comments, all related to your comments about science. I'll read you Vince Young. I agree with the benefits of seeing science, but many people have been trying to reshape the entire academic training environment. We basically have to tear down everything and start over. Not sure how to do this. And then another person asked whether you could speak a bit to what we need to change regarding peer review, preprint, and publishing process, and any suggestions for creating a system of science that is seen as inclusive and collaborative at all levels. So, Nevan, you clearly touch a nerve here. So why don't you tell us what you think?
Yeah, I mean, it's these are all fantastic questions. These are ones that I've been racking my brain about over the last several years, and even more so over the last 6-7 months during the pandemic. And it is, I guess I am suggesting is to rip the whole system down and start over, but you can't do that, right? I mean, you got to take it in a stepwise fashion. And I guess from my end, okay, what can I do? Well, what I can do is lead by example. I could say, okay, look at what you can do when people work together. Look how fast you can move when people go to work together. And I know we're getting a lot of interest. People are looking at what we've done and say, well, maybe this could work at the end of the day. And maybe we should try this in these different disease areas. But I think really, as we discussed earlier, a way to go forward is to change the reward system. And again, focus your attention on younger scientists. Make sure if credit is given, make sure that they get it at the end of the day, and reward groups instead of individuals. Why can't HHMI give out a grant to a group? Why does it always have to be an individual at the end of the day, right? I mean, I think you could start to make these smaller changes with these very prestigious awards as well. And when we all stood on study section, instead of saying, why are there so many authors on this paper? Why don't you say, this is great. Look at all these people that have worked together. They should be rewarded. You know, so I think the mindset has to change with the scientific community. And I think that would go a long way to making differences across the board.
Well, Nevan, I think that we reached the 10:45 point in which panel.
Oh, Arturo, there's one more thing here. I'm curious about the decorations on your lapel. What is your message here? So this is from Canada. So it's a Canadian pin, obviously, but this is a poppy for Remembrance Day. I think in the States, you guys call it Memorial Day. That's tomorrow.
So I believe that's a Flanders poppy, right?
That's right. Very good.
The Flanders fields, where one of the great useless slaughters of World War I.
That's right.
The Canadians suffered tremendous casualties. Was it Vimy Ridge?
That's right. Very good. So I'm glad we can end on that.
Well, it's good. Yeah. So I will turn it over. I believe Susie will now introduce the panel 1.
I think this symposium is off to a really wonderful start, and thank you, Nevan, for your very inspiring keynote address. We'll now begin with the 3 panel discussions, and I'm Mary Estes of Virology from Baylor College of Medicine, and I'm going to briefly introduce the topic of panel 1. And the title of our panel is Elevating the Role of Rigorous Scientific Research in the Post-COVID Environment. This important topic, which was touched upon in the keynote address, has been designed to address really the following objectives. First, to re-emphasize the importance of scientific research. Second, to explore new scientific areas that have emerged from the pandemic. Third, to demonstrate the value of science to gain public trust. And last, to deliver clear scientific information to the public. We expect this discussion will help us understand new models of collaboration across academic, industry, and government institutions, as just discussed, that can continue to facilitate solutions to the ongoing and future crises. That it will help propose to maintain how we can maintain the appropriate research budgets going forward so we can be prepared to overcome the current crisis and handle the new ones, and allow us to better understand how to engage the public so they can learn about public health and have confidence and trust in science. I'm now delighted to introduce Dr. Vincent Racaniello, who will moderate this panel. Dr. Racaniello is a professor at Columbia University who's done laboratory research on viruses for over 40 years. Following up on his belief that scientists must communicate their work to the public, he has co-authored a virology textbook, distributed videocasts of his virology lectures online, written a blog about viruses, and produced podcasts on viruses, parasites, bacteria, evolution, and immunology. During the COVID-19 epidemic, he's become a reliable source of information on the SARS-2 coronavirus and the disease that it causes for thousands worldwide. This makes him the Earth Virology Professor and an exceptional person to introduce and lead this panel of eminent speakers. Please join me in welcoming Dr. Racaniello.
Thank you, Mary, and thank you, Arturo and Susie, for having me here. I'm looking forward to a nice discussion with our panelists. Let me introduce them to you to start. Peter Hotez is Dean of the National School of Tropical Medicine and Professor of Pediatrics and Molecular Virology and Microbiology at Baylor College of Medicine. He's also director of the Texas Children's Center for Vaccine Development and Texas Children's Hospital Endowed Chair of Tropical Pediatrics. He's an internationally recognized physician scientist in neglected tropical diseases and vaccine development. Most recently, both as a vaccine scientist and autism parent, he's led national efforts to defend vaccines and serve as a champion of vaccines, going up a growing national anti-vax threat. Welcome, Peter, to our panel.
Thank you.
Geraldine Miller is a senior director on the AI for Good team at Microsoft, where she focuses on health strategy. Geraldine's business acumen is supplemented with a deep technical knowledge base, including work as a software developer and systems administrator on a variety of platforms using many software languages and tools. Geraldine is a Time magazine author on the topic of artificial intelligence in healthcare and a speaker on the topic of health equity and AI. Welcome to the panel, Geraldine.
Thank you.
Tara Smith is an infectious disease epidemiologist at Kent State University College of Public Health, with 20 years of experience studying the transmission of zoonotic disease primarily in rural communities. She's worked on various aspects of COVID-19 epidemiology and response, working with communities, churches, and migrant workers to aid in understanding and prevention of infection. She's also written over a dozen articles on the pandemic for popular press outlets and has provided hundreds of interviews for outlets from local to international. Welcome, Tara.
Thank you for having me.
And Susan Weiss, professor in the Department of Microbiology, Perelman School of Medicine, University of Pennsylvania. She's worked on many aspects of coronavirus replication and pathogenesis over the last 40 years, making contributions to understanding basic biology as well as organ tropism and virulence. She's worked with marine coronaviruses, MERS coronavirus, and most recently SARS-CoV-2. Her work for the last 10 years has focused on coronavirus interaction with host innate immune pathways and viral innate antagonists of double-stranded RNA-induced antiviral pathways. Welcome, Susan.
Thank you, Vincent.
You and I have 40 years together.
Yes,
80
we do.
years altogether. Wow. When people say that, I feel old. We have three separate issues to chat about, and people listening in, the audience, you're welcome to put questions in the Q&A, and I'll look at them and sprinkle them amongst our discussion. The first one is all about basic science. And I want to start out by asking, how has our investment in basic science allowed progress so far in understanding SARS-CoV-2 and COVID-19? How has it guided development of diagnostics and therapeutics? And I want to start with you, Susan, because you've been in the field a long time and have a good 20,000-foot view.
Yeah, I want to go back a little bit in time, or maybe a lot in time, to the beginning of the coronavirus field, just briefly. I won't bore you too much, but back in the 80s, when a lot of the sort of molecular biology on coronaviruses just began, I think we worked on it, a bunch of us worked on it because it was a really interesting field. Just there was a lot of basic virology to be done. We knew this virus had a large genome, and it had an interesting and different method of replicating its RNA, and it had encoded a lot of non-structural proteins. It was really interesting. Plus, there were interesting animal models to be studied, particularly for mouse hepatitis virus. So this field really began, I would say, from a vaccine development aspect, and also from just a basic virology. And there was really a lot learned during, I would say, the 80s and the 70s, but mostly the 80s and the 90s. And I'm not going to go through too much of it, just to summarize, we learned a lot about the spike proteins, about the fusion protein, that they had two subunits, they had a receptor binding domain, that some spikes were cleaved by furins, some were not. We learned about cell-to-cell fusion. We really learned a lot about the biology of the spike protein. And also, it was really pretty early on that we started to get clues that these spike proteins could mediate entry by two different pathways. So all of this kind of very basic work on the spike, I think, has really taught us a lot and prepared us for vaccine development, and also just for thinking about antivirals that might inhibit entry. So I think that's been really important. We also learned about the different genera of coronaviruses that for MERS and SARS, for example, probably would not induce cross-reacting antibodies and things of that sort. So we learned a lot, I think, towards vaccine development. We also learned, and plus there was a lot of vaccinology done for the animal viruses, for infectious bronchitis virus, bovine coronavirus, the porcine viruses. And then just in addition, I think antiviral research was also greatly benefited by this early research, where we learned that these viruses encoded two proteases that were from different classes, the papain-like proteases and the 3C-like proteases, from 3C named because they were similar to poliovirus proteases, something that Vincent knows a lot about. We also learned about that these viruses made these huge, long polypeptide and encoding multiple other enzymes that are all Nevin talked a little bit about this. This is like 40 years later, but we defined the basic biology, what these enzymes were. The proofreading enzyme becomes important, so we know it can attenuate mutations in that enzyme, can attenuate virus. There's an endonuclease that prevents accumulation of double-strand RNA, deubiquitinase, all kinds of really interesting domains. And this was all worked out, I would say, pre-SARS. A lot of it was pre-SARS. And then the third thing that I think was really important was all the sequencing, that we knew a lot about these viruses. And when SARS appeared, it was really identified rather quickly. And I'm sure that's really a result of all the basic biology that was done. Ralph Barrett cloned it in a record time. And all of this really accelerated to MERS and to SARS-CoV-2. So I just want to emphasize that all of us that began working on these viruses, it was always difficult to get funded. It was always difficult to rationalize why NIH should fund a mouse hepatitis, a murine virus. And I think now we can understand why that's really important. The one other thing I think that came out of all this is the virus-host interactions, which we knew quite a while ago that these viruses really shut down interferon pathways. And now that becomes important in that we see that people with dysregulated interferon are more prone to a more severe COVID-19 disease. So those are just some of the examples of how I think, even going back further than working on the current virus, that we don't know what the next virus is going to be. So somebody's going to write a grant on some obscure virus that nobody cares about. And we don't know, is that really going to be important 20 years from now? So that's my thoughts on that topic.
I think it's still difficult today to get funding to work on mouse viruses or any virus that doesn't cause human disease. It's getting harder and harder. But this shows that, the coronavirologists prevailed, even in the 60s and 70s when we didn't think they caused much disease. And I'm not sure we'll change, but we'll see. Maybe we can talk about that. Peter, in terms of vaccinology, what set the stage for maybe having a vaccine in a year, an amazing feat?
Yeah, I mean, just to build on Susan's comments, we were not virologists. We're not, in fact, we never were virologists. We moved to Baylor about a decade ago, and because Mary Estes was at Baylor and was helping lead this incredible department of molecular virology, I thought, when you make a move, it's always good to learn new things. So I thought it'd be really exciting to dip our toes into virology. And then we got approached by a group at the New York Blood Center, Shibu Jiang and Lanying Du, who had been working on coronaviruses for quite a while. And they were getting frustrated because they had a very good idea for a vaccine concept, yet they couldn't attract any vaccine developers to work with them on it. So even though we were working on parasitic disease vaccines our whole lives, we started partnering with them. We wrote a grant to the NIAID, NIH, and it got funded. It was a pretty large grant to develop receptor binding domain vaccines for SARS and MERS. And they were really effective. One of the things that they had noticed was if they used the full spike protein or an activated virus, they got a fair bit of immune enhancement, a lot of eosinophilic infiltration into the lungs or liver sometimes. Whereas if they truncated it to the RBD, they were able to achieve higher levels of virus neutralizing antibody without the immune enhancement. So we began scaling up production of those, and we actually manufactured them under GMP, under good manufacturing practices at Walter Reed, and worked with Ken Cheng's group at UTMB Galveston. He had developed a mouse model using a transgenic mouse model of the ACE2 receptor, also had mouse adapted virus strains, and gave really great protection. And then this is so classic. We had it manufactured and then nothing. For both SARS and MERS, we couldn't attract any investment or funding to move it into the clinic, and that was very frustrating for us. And then when we saw the sequences of the COVID-19 virus, the SARS coronavirus type 2 up on bioRxiv, I looked at it and I said, this is close enough. We should be able to hit the ground running doing the same thing with COVID-19. And we took a two-pronged approach. We took the old SARS-1 vaccine out of the freezer to see if we can get heterologous cross protection. And with Lanying, we were able to show, indeed, we get some cross neutralizing antibodies. But everybody we approached with this idea absolutely hated it. They just we could not attract any interest at all in moving the SARS-1 forward. But we were able to use all the techniques that we had used for SARS-1 and applied for SARS-2. And it was simple things like, but it still took a lot of time to work out. So for instance, there's an N-terminal asparagine, which is heavily glycosylated in yeast. So we found if you express it without the N-terminal asparagine, the structure stays the same and we get really high levels of expression, and then it's super immunogenic. Or in the case of these RBDs, there's a free cysteine in there. So there's an odd number of cysteines that formed intermolecular disulfide bonds, which from a production standpoint is something you can't control. So we engineered that out. So we had done all those things with SARS-1 so we could hit the ground running with SARS-2, and we moved really fast. And now we're getting great levels of virus neutralizing antibody and working now in India with Biological E, a BioE company in Hyderabad that's scaling up to make 1 billion doses of it. So that's very, very exciting for us because we never made 1 billion of anything before. And but it builds on all the work, that Susan had done and the coronavirus biologists have done. So, people start thinking it's a miracle how quickly we were able, that we could move all these vaccine concepts into the clinic. And they fail to realize it builds on 30 or 40 years of basic research. It just didn't happen in a vacuum. And that's a hard thing to explain to people, but something that's really important. And, every chance I get, because I do a lot of things on TV, I remind the American people why it's worth spending $36 billion a year on the National Institutes of Health. This allows us to do these kinds of things. And I think people are starting to get that message now and hear from scientists like they never have before. And And I, say all of the concepts of the vaccine, even though, some of them are mRNA vaccines or adenovirus vaccines or nanoparticles or our recombinant protein vaccines or even inactivated virus vaccines, they're pretty much all operating on principles that were worked out over 40 years around the spike protein and the importance of the spike protein or the receptor binding domain. So it really is in some ways an old school problem in virology that had been worked on for decades, and we were able to build on that.
Yeah, I think we'll talk later about how to get that message across, but I think this is our moment to really emphasize that what has been done with basic research in the past and how it makes us more nimble today. And you speak to this problem in getting vaccines for things that are not of interest. That's a tough one too, because we were really rushing to catch up, and I don't think we had to. I think we should have been better prepared. But I'd like to hear Tara's view on this. I'm sure you're thinking about this a lot. How are we building on what's come before?
Yeah, so for mine, it's a little bit more applied because we're looking at transmission. And so we have some cohorts that have already been established, not at my institution, but using others that are building on cohorts that have been established looking at influenza or other infectious diseases. And they're using those now to study coronavirus. So from my point of view, it shows the importance of basic surveillance, not only in humans, but of course the zoonotic aspect as well, of years of studying zoonotic diseases, of looking at bat pathogens, even if they're ones that potentially may have very limited potential to cross species. You only need one successful transmission, right, that you can then spread in the human population. So looking at all of these different aspects of surveillance, both within human populations and at that human-animal interface, and having those in place and having people with that a type of expertise, I think has been really critically important here in understanding species jumps in general and coronavirus in particular.
So Geraldine, you have a different perspective. You're from AI and software, and yet I think you might be seeing things that were developed in the past that are helping us now.
Yeah, and so, I want to share a little bit of history from a Microsoft perspective. So, Microsoft has had a research subsidiary since the early 90s. So this concept of taking, state-of-the-art computing methods and applying them to domain-specific problems to solve world problems isn't something new for us. I'm on a team right now over in philanthropies at Microsoft where we focus on bringing artificial intelligence to the world's greatest problems. And this covers everything from investments in Earth to accessibility, and more recently, a set of investments in bringing artificial intelligence to the domain of healthcare. So we launched this AI for Health initiative in January of 2020. I can't say that it was in complete absence of knowledge of SARS-CoV-2. A little back channel stories. I can remember the eve of the launch, our data scientists coming with some visualizations they had done on the progression of the disease, and they were very scary curves from an epidemiology perspective because they were exponential curves. But we went ahead and launched our AI for Health initiative, and 3 pillars in that. One is focusing on health equity, global health insights, and then a category that we called Quest for Discovery, which is really basic scientific research. Over the course of the next few weeks after that January launch date, it became obvious that COVID-19 and SARS-CoV-2 viral infection rates were something that were here to stay, at least for some time. So we took a quick pivot and launched a call for proposals to support research in the area of COVID-19. So as a technology company, we're really focused on bringing artificial intelligence, data science, and analytics to these problems. And so our focus is on enabling researchers with compute and with data science from a collaborative standpoint. And so in our COVID-19 set of work, we have several different topics or focus areas. And one of those is still basic scientific research. So we continue to support this through the application of high performance computing assets, and then also artificial intelligence and expertise in that area. But it's not just Microsoft. We're not doing this in a vacuum. If I step back and look at what's happening in the industry, by example, in the US today, there is an initiative, a consortium called the COVID-19 HPC Consortium, where there are cross-domain and cross-sector individuals coming together to offer compute resources for COVID-19 researchers. And this covers everything from your large cloud vendors, so Microsoft, IBM, Google, Amazon Web Services, to DOE-sponsored infrastructure. There's also some international members as a part of this consortium. And really this is geared at it again, offering up these compute assets to help solve some of these challenging problems for COVID-19. There's also initiatives that are stepping away from the current pandemic and saying, what do we need to learn about how we come together and organize, and what are the things we need to focus on going forward? One of these is a challenge or a coalition called the Trinity Challenge that is led by Dame Sally Davies in the UK. And this is looking at, identification of disease, how we respond to disease and recover, and it's a challenge. So researchers are coming with proposals to the challenge, collaborating together, and then this is an example of one where there's prize money as well associated with the winners of the challenge. So a couple different ways that different entities cross-sector are coming together to address the challenge of, how do we solve this problem that we still have some fundamental basic research that needs to be done.
Speaking of the future, this won't be the last pandemic, obviously, and many of us talk about how we should have been better prepared for this one. So, what once we have this under some sort of control, what should we be doing to be ready for the next one? Can we do anything different, or when this is done, will we go back to the way things were before? Susan, I know you have some ideas about this.
Well, one very specific thing I think is to continue to support antiviral drug development, because that's going to be really important for the next virus that doesn't respond to whatever vaccine is developed for SARS-CoV-2. So I think that's really important. And that means continuing to understand all these non-structural proteins that the viruses encode, and really continue. I know that some people like Mark Dennison were working on antivirals in between MERS and SARS, but maybe not getting as much support as they could have. And I think that's a very practical thing that can be done. In addition to, I think also continuing to identify what Tara said, continuing to identify viruses in bats and other species. So we really know what's out there and what we can expect. We can know, I think one of the studies I read suggests that SARS-CoV-2 really could have gone directly from bats to humans, and that there are viruses in bats with receptor binding domains that may interact very well with human ACE2. And that may be true for other coronaviruses. We don't know what's out there, or other non-coronaviruses. So I think that surveillance is really important going forward. Those are two things that I can think of right away.
The problem is, can we get the money to do it? As you know, surveillance programs were curtailed by this administration. And we do hope it'll change. But the problem is, if there isn't a virus, there tends to be little interest in supporting work on it. I mean, what do you think about that, Tara?
Yeah, it's problematic. And again, as a bacteriologist, I would say that we need to put out a broad net. I study in my day job antibiotic resistance, which also is a coming threat, a current threat, and one that is also difficult to get funding for. It is surprisingly difficult. So I think we need to look at this animal-human interface, not only for little specific niche viruses or bacterial species, but to really broaden it. And we have a lot of tools to do that now. So it's really kind of surprising to me that we haven't put more money and funding and really importance, I guess, in that area where I think we have the ability to accomplish so much and to get so much data from relatively small, potential studies.
Yeah.
Could I just say
of course,
something?
go ahead.
It'd be really nice if NIH put out an RFA for basic research on viruses not known to cause any diseases, just to have some, instead of an RFA for Zika and everybody runs to Zika, an RFA for SARS-CoV-2. And I don't know how many grants were written on SARS-CoV-2 in my institution alone, but when SARS goes away, is there going to be, who's going to be left to work on this virus and other viruses? So if NIH could put some effort and some money into that, it would be really, really helpful going forward. But like you said, people forget, and then it's tough.
We've had, SARS-1 in 2003. We've had MERS in 2012. Now we've got COVID-19. Guess what? There's a pattern here. All right, we are going to see a new coronavirus of serious disease consequence every decade now. That's our new normal. And, the flu people have done this with some success. They've put together a fair bit of infrastructure, both nationally and internationally, around flu detection, avian flu, swine flu detection, and also countermeasures as well, looking at universal flu vaccines. So are there lessons learned that we can apply to coronavirus, and looking at universal coronavirus vaccines? What would that look like? Could we combine antigens for all the existing coronaviruses, and would that give cross protection that we could bottle and give out next time? So I think that's going to be really important for the virology community to look at, and use maybe flu as a model and look at what worked and what didn't work for flu and apply it to coronaviruses, because I think now the world gets it. They didn't get it after SARS-1 or after MERS, but maybe this time, this one's the trick. Although, it's always like a little kid's soccer game, when you watch little kids play soccer, when I used to watch my kids, the ball would go off in one direction and all the kids would run after it. Then the ball would go over there and all the kids would run after it. No one would stay behind and play defense. And that's what's happened with the pathogen community, unfortunately.
And also just to emphasize, each one of these epidemics and each one of these viruses has been somewhat different. So I do think, again, it's important to understand the basic stuff so we know what it is when it arises. I mean, MERS and SARS are pretty different in their sequence of spike, and the way they've behaved is really different. So
I think we have to somehow get to elected leaders and make them realize that, and if they don't get it by now, that these viruses are not just health threats, right? This destroyed the global economy. It became an international security threat. So all of the funds that are put in place to counter the development of nuclear weapons, we need to start doing the same thing around viruses. And I think this should be a topic for a future G20 summit, because I think it should be looked at all the G20 leaders, which control 86% of the global economy. They're the ones we need to target.
So Peter, I'm gonna pile in on your example of children's soccer. We used to call that bunch ball, right? Where everybody bunches around the ball. And I do agree that's kind of what we're seeing right now, with everybody chases what the current health threat is. But I also agree that it's not just viral threats, right? There are other potential global health crises looming, and so what we need to do is understand, what have we learned from this current pandemic that would have us more organized on our soccer field of life, and where we're each playing in our particular roles, and we have well-defined roles, and we come together as a team. And so this collaboration aspect, I think, is really important. I think it is, in some areas, a highlight of what happened with this pandemic was to see the cross-sector collaboration. And so I think one of the learnings is how do we enforce that type of behavior coming out of this, and then also stepping back and looking at it from the perspective, how do we make sure that we have the ability to spin up what I would call almost like the special task forces for the next global health threat, right? How do we bring people together quickly? how do we have the fundamental infrastructure to enable this from the standpoint of things like data sharing, right? The ability for people to work together. And so there are some things I think we can learn out of this pandemic, and a lot of it is about how people collaborate together in the name of science, and also how people come together from an international perspective as well, which I think was something that was done really well in the current pandemic.
Yeah, I mean, I think, what we saw with this is it brought out the best and worst. The best in terms of, putting up papers on bioRxiv now or medRxiv is almost routine now, whereas before it wasn't necessarily. I think it's almost universally accepted now by journal editors that it's okay to put your data up on bioRxiv. So people were sharing, and a lot of the information we got were Chinese scientists were putting up their data on bioRxiv. So when the accusations are the Chinese weren't transparent from the scientific collaboration perspective, I found that what they were, they were. But then it also created a dark side too. For instance, in Houston, they shut down the Chinese consulate in Houston, allegedly because they were spying on vaccine technology. And I got asked by a journalist about it. I said, they don't need to spy. And I mean, all they needed to do was send an email and a calendar invite, and we would have told them what they're doing. And that got published. I don't think Baylor Legal was too happy about that. But it's, so I one of the better sides of this, I think the scientific community really stepped up in a huge way in terms of collaborations and being transparent about data. Everybody was driven to do good and wanting to, sure, there were competitions as well, but I think generally speaking, the scientific community stepped up in a big way to want to work together to solve this problem.
Another area that we wanted to touch on has to do with new technologies that have been developed and have been brought to this problem. And Geraldine, maybe you could touch on AI and how it can be used in life sciences research.
Right. Yeah. So, it's interesting because we're seeing such advances in the way that AI is being used generally in the areas of health. So a couple of ways, right? One is about the gathering of data and insights, right? And so this is really information about people's safety, economic impact, the effect of policies, right? And although that seems far removed, I do want to, make a call out to the comment earlier about this just isn't a health crisis, it's a global economic crisis. So really understanding how the data helps drive and shape policy and efficacies of policies is important. We've seen AI applied to things like treatment and diagnostics. So enabling research to further the development of vaccines, diagnostics, therapeutics. We saw a lot of work in that area in the HPC Consortium, people using different molecular dynamics tools and modelings, quantum mechanics and molecular mechanics simulations. In the area of health, it's also about allocation of resources, and that's not just from an economic standpoint. How do you use AI to do things like predict pathogenesis of the disease? And so you can determine, for example, how you allocate scarce resources like ICU beds or perhaps ventilators or other PPE. And then of course, back to this basic scientific research. So many ways that AI can come to bear in the current pandemic and going forward as we look to global health crises.
Yeah, I have seen many studies where the quantity of data generated and examined is far more than you could just, when I trained as a PhD, you could look at it and see what's going on. You can't do that anymore, right? You need to have algorithms. So we need to have a real interface between, scientists like me and Susan and Tara, who are trained doing simple things and using algorithms to analyze them. And I think an outbreak like this really brings those groups together, right?
Just a quick comment. I'll give an example of this. We did some work with UCB to support them. They had a group of scientists who were coming together to participate in the COVID moonshot. And so we were able to work with them collaboratively to spin up in in a few days, tens of thousands of compute nodes all over the world in our Azure data centers. And this is something that, had they run on their own internal infrastructure, would have taken 6 months. So just the capability to put raw compute power and models on some of these problems can be hugely beneficial. And this is a cross-domain collaboration, and I want to emphasize the value of that, right? Bringing together people from disparate backgrounds, computer scientists with biologists, with medical experts. And not only does that help reduce bias in terms of in terms of the AI and the models that are built, but there are really some great things when you have this cross-domain collaboration.
Yeah, I have to say that you have to break those silos, as our keynote speaker said. I had a conversation with someone from Google really early on, and they said, "We have all this computing power. How can we help you?" And I said, "I don't know. Go talk to someone else. I have no idea." But what have there been other technologies developed during this pandemic? Susan, is there anything that caught your eye?
Not for me. I'm with you, Vincent. Actually, the people from Google asked me the same question, and they offered me their computer help, and I really didn't have anything to need at the time.
Yeah,
well, the way I do.
There are clearly people who do, and I think that's really important that they have bridged it. You and I are the dinosaurs, I guess,
right,
and
we are
not the future. But
I think a couple of technologies that got accelerated. Well, the adjuvant technologies. It's allowed us to move along some adjuvants. Mm-hmm. Eventually, we're going to go into the clinic anyway. I think this kind of pushed it along quite a bit. So that I think has been really helpful. Also, the new vaccine technologies, mRNA vaccines, DNA vaccines are all moving along, but this definitely took it to a new level. People say we sort of went overboard with that. We veered all of a sudden, we relied exclusively on new and untested technologies, and maybe we should have balanced it more with some traditional technologies like ours, which were accelerating in India, but not the United States. And maybe Operation Warp Speed should have balanced it out a bit more without rushing after, mRNA and adenovirus, which we don't know anything about the durability of protection or if there are any going to be safety issues downstream, or really not even knowing what the immunogenicity was going to be until pretty late in the game.
Yeah, that's a good point. In terms of vaccines, we have really avoided the traditional Salk-Sabin style vaccines offer new
Well, those are there too. So, for instance, the Chinese have 2 different inactivated virus vaccines on alum, and India, Bharat in Hyderabad is making one. And you know what? They're pretty good vaccines. They work.
I also wonder about the attenuated vaccines. I know that everyone's afraid of reversion and stuff, but there are so many ways to cripple coronavirus, ways that would be really unlikely to revert, like deletions and mutating a bunch of the antibodies.
Yeah. There is a company in upstate New York, right? Codagenics, that's doing that. I don't know how they do it, but that's.
It just seems to be like you're presenting all the proteins in their normal way. It just seems like a good approach to me.
I think the development path was just longer than making an mRNA or a protein, right? And that's probably what won out.
I think the problem with that is I think there are upfront gains, right? You make a piece of mRNA pretty quickly, but the problem now is you got to deal with the production issues of the lipid envelope, and it requires, as we all know, RNA is pretty unstable. So now you have to deal with minus 94 degrees Fahrenheit frozen temperatures, whereas older vaccines like ours, even though it took us longer to get into the clinic, we can move pretty quickly. So I think it'll kind of balance out at the end. But we'll get better at it. We'll get better at RNA technology next time around. So I think this will definitely open the door for lots of new vaccines and new delivery mechanisms also.
I've volunteered my extra minus 70 if anyone needs it. Some doctor's office, they can have it. I wanted to ask Tara, I hear people lamenting about the lack of opportunities to do field work in virology, in microbiology in particular. I think that'll change.
Oh, I hope so. For me, fieldwork has been the best and most fun part of my career. So going out and taking samples and enrolling individuals and learning to interact with people in the community and talk science with them at the same time, I think is a great opportunity. And we're seeing people do that, even though we don't have necessarily a lot of field work going on. There are a lot of people getting, work as contact tracers and things like that. So learning how to do some of this even over the phone to talk to people about their infection and their exposures and things like that can be a way to kind of break into some of that as well. So I think there are some opportunities here, and I do hope they will be going forward in the future, because going out and swabbing people, swabbing animals has always been a great experience for my students. So hopefully we'll see more of that.
So in the last 15 minutes or so, let's talk about communicating science. And I think there are two audiences, right? There are the general public, but then there are also policymakers and lawmakers who should be informed. I'm a big fan of communicating science, and it's clear why it's important. It's been very clear in this outbreak. But I'm curious as to, I think we have a sense of the answer to this, but I'm curious about how your public engagement has changed during the pandemic. Geraldine, have you seen a change in your engagement?
Yeah, and so, when I look at the work that we're doing in AI for health for COVID-19, we have this category of research that we're doing called dissemination of inaccurate information, right? And so this kind of has two factors. One aspect of this is really what you see happening, for example, on social media, right, where people take information and then they populate it or they otherwise spread the information without the pause to think about the accuracy or the provenance of the information. Where did it come from? Can I trust this information? So we have some active research going on in that area with people who are looking at this from a behavioral standpoint, and people who are looking at it also from the standpoint of, how do we do things like trace provenance of information and do that responsibly? So that's kind of what I would call the consumer or the general populace side. But there's also some work going on, a lot of work going on the academic side as well. How do people from the corpus of academic literature extract information, and how do you use artificial intelligence, machine learning, and different tools to be able to get at the information that you need? And also, how do you do this in a somewhat remote environment? We have one project that we're doing with the University of Notre Dame, where they did a bunch of work on the core D19 data set, and they're enabling faculty and students to take this data offline in what they're calling like a virtual study carrel. So enabling new methods to be able to get at this research and kind of understand this research. So I think there are very definitely opportunities from both a public standpoint and from a research standpoint. And I also think that there's this concept of what I will call literacy. I think the general populace, there needs to be a certain level where people can understand some basic scientific principles to be able to make decisions. Not everybody has to be a biologist, but we need to understand the impact of our decisions. When I was working in genomics, I saw this as a huge problem. There's this concept of genomic literacy as well, and so I think we need to make sure that we're encouraging that for the general population as a whole.
Do you think it's possible one day to use AI to fact check social media?
I think that there's a lot of work going on in that area today, and so that's the hope. Right?
Yeah, because having hundreds and hundreds of people do it seems crazy, right? And then you're still subject to their interpretation. Susan, how's your engagement changed?
Well, at the beginning of this, my life took a huge change. I was being called by newspaper people nearly every day. I did a couple of 30-minute in-depth interviews that were really kind of fun to do, and from a local news person and from some more academic places. So I just saw so much interest that I felt like it was my duty in a way to go out and tell everybody about these viruses. They weren't something that just appeared out of nowhere, that there was actually a long history of them. And I got really, really tired of doing that, I have to say. But I think it was really important. And I also noticed around me that people, both scientists and lay people, coming up to me and saying, hey, viruses are really interesting, just from an intellectual perspective, not only from COVID-19, but people wanting to know more, even in my own family, wanting to know more about viruses. My son, who's totally not a scientist, was, really taken by all this. So I do think that naturally people have become more interested. I mean, to see my virus that nobody cared about on the front page of the Times, pictures in Newsweek, pictures everywhere, is really, truly ridiculous. It's really amazing. And I think, Vincent, your podcast, your TWIV is exploded. Everyone I tell about it, and they listen to it, and they tell me how much they really enjoy it. So this has been an opportunity, I think, to make virology something cool, something that people want to hear about. And I mean, I guess the challenge is, how do we keep this going? How do we keep people, continuing to be interested in all this?
Yeah. No, I've noticed so many people say, wow, I didn't realize viruses were so cool. I said, I've been doing this for 40 years. Where have you been, right?
Of course they are.
Tara, what have you been doing?
Yeah, I mean, mine has been mostly the same, just kind of like Susan said, just more.
Mm-hmm.
I think it's really a challenge for, reporters to identify people who can not only talk about the science and have expertise in one of the areas of virology or epidemiology or molecular biology or whatever the particular reporter is looking for that day, but they're also able to translate that for audiences. So I think that's difficult for some reporters to find. So I do appreciate that ASM has put out some resources for some of that, but I think a lot of reporters are still looking for those individuals. So I've tried to serve that whenever possible. And then I've also recommended, of course, many dozens of others throughout the pandemic when it's in areas where I just, it's not my area. So I've been able to build up a good network, I think, for that, which has been nice, and that has changed a bit. But one thing I do want to just talk about here briefly is how all the speakers have talked about breaking out of silos. And usually that means across, different areas of microbiology or molecular biology. But I also want to remind everyone that we need to include our colleagues in the humanities as well. I mean, so people who do communication professionally, people who research conspiracy theories and, data analysis, who do social media, analyses of how some of these things spread on Facebook or Twitter or whatever. Historians who have studied different pandemics that we can bring into this conversation. So I think this has to go beyond just the science as well. Obviously, we're seeing a lot of the problem in communication here is on behavioral aspects, which we have, health educators and behavioral scientists who are trained in helping people change their behaviors that, as microbiologists, we may not, that's out of our depth. So I think we need to make sure to, send that shout out to our colleagues and reach out to them across the humanities as well in some of this, because they are trained for some of this, and we may have the scientific expertise, but we don't always have that communication aspect down pat.
Peter, you've always been a communicator. What's changed in the past 8 months?
I think one of the big changes, Americans started hearing directly from scientists for the first time. In many cases, the first time they've really heard from scientists. And we're no longer invisible. And for me, it took it evolved in a very interesting way. I think initially I was out there trying to fix the terrible communications coming out of the White House coronavirus task force. It was mainly because it was some communication specialist told them they have to talk to the American people like they're in 4th grade or 6th grade. And it was so simplified and dumbed down that you couldn't even figure out what exactly they were saying. And I think it came out of an old school idea out of communications that the American people can only understand things at the 6th grade level. And that was sort of pre-internet, before I think the American people are a lot more sophisticated than that. So actually sitting down, actually taking the time and getting the time on air to explain the science, people loved it because for the first time, they were actually hearing the underlying assumptions why you would make statements about masks or contact tracing or social distancing. And that proved to be really important because then as the science shifted, even though what you said might have been incorrect 4 months ago, you could go back to, yeah, I said that, but remember I gave you the following assumptions and now this assumption's changed and this is why there's been a shift. And people like that very much. And then it took a very dark turn over May into June when the Trump administration decided they weren't going to do anything at the national level about COVID-19. And instead there was a deliberate disinformation campaign, right? And finally they brought out Scott Atlas, and the disinformation campaign was to deny the severity of the epidemic, to attribute the COVID deaths to comorbidities, not to COVID-19. There was all the fake herd immunity ideas that herd immunity was going to occur at 22%. And they even, got the Koch brothers to fund this Great Barrington Initiative to do that. And then there was discrediting of masks, and so that became my role was to basically call it out each time and point out that this was not an accident. This was a coordinated disinformation campaign, and it was really important, I think, in the 2020 presidential election. And then I had to avoid sounding like a crazy person because then you had Russia and Russia Today, filling our internet with disinformation, and then you had the Murdoch media empire. Wall Street Journal was putting out op-eds discrediting masks, and I started sounding like a crazy person who throws things at the television talking about the Russians and the Murdoch media empire. So I had to figure out a way to say that and still sound credible. But it was scary because in academic health centers, if scientists speak out at all, they're warned by the Office of Communications. Remember, just stick to the science, just stick to the science. And you do that as much as you can, but what do you do when the White House has deliberately tied this to a political agenda, like a whole disinformation campaign as a strategy to get reelected? There's no way to disentangle it. And this created a real internal struggle. And I remember back in May, my wife Ann said, I can see this is really bothering you. You're just going to have to let it rip. And I did. And it's a scary place because it takes you into some very dark spaces. And you can imagine the mail and the email and much less social media. And there's no roadmap for this, right? Because this is so unprecedented in modern American history. And, I'm still winging it going along. But I think the message is there was an assault on science. It was a deliberate assault on science. And this idea that scientists, have to stay in their lane and can't speak out, I think this is telling us we need to pivot and shift. And I think we need to do a better job in our doctoral and postdoctoral training for actually bringing in science communication and public engagement into our training. Not everybody wants to do it. And it is, it can be a distraction. It can take a lot of time. But in this case, it became essential for national security to do this. And I think if, a lot of young people love the idea. I mean, the commitment to public service for young people is at an all-time high. And this would be one manifestation of that, bringing into the DNA public engagement, science communication. That's a shift because, when I was in New York, when I was getting my MD, PhD, the message was you don't talk to journalists, you don't talk to the public. That was seen as a form of self-promotion or grandstanding. And all those ideas were put in place before something called the internet came along and social media. And I think it's all changed. And so podcasts have become so important, like what you're doing, Vince, and others. And so I think this is going to be we don't really have an organization or a mechanism to do this at a high enough level and an ambitious enough level. I know ASM has worked really hard on this front, but I think it needs to expand and really embed it into the DNA of our profession.
Sarah, what do you think? Tara, sorry, what do you think of this idea of having it part of, say, PhD curriculum?
Oh yeah, I've been arguing for that for a while. That at least for students who are interested, have some kind of training in there. We have PhD programs that focus on grant writing, which of course is a different form of science communication. So this could just be an addition to that. I would also add that when I have publications in the area of science communication, they really don't fit in any of the ASM journals either. So if we had some niche where we could send those, I know Peter's also published a lot in the area of science communication, and I would love to put those in ASM journals. So if there's some way that niche could fit into either one of the established journals, or I don't know about it.
You're absolutely right. I have to plead and cajole editors to take that, and they have to do it quick too, because sometimes these things have to happen in real time. I think the other problem is the academic health centers don't like it. They want to control the message. They're very risk averse. They want to protect the institutions, and having their docs and scientists speak out about combating disinformation, calling out the White House, or speaking out on social justice, it doesn't fit. And so it's not something that's general. I mean, people at Baylor and Texas Children have been really good about letting me do this, but a lot of places they really clamp down, and even then, that's not something that goes into your you can use for career building, right? I mean, I have to fill out an annual evaluation form, right? And what they want to see in the annual evaluation form, they want to see my grants and my papers, and they want to see my grants, and they want to see my papers, and they want to see my grants, and they want to see my papers, and the idea that there's not even a place on my annual evaluation form for the books I write. I mean, I like to write, and there's no place on that, much less bed pieces or the stuff I've done on media, and certainly not any kind of social media. So I think we've got to find a way to encourage that and not see it as something to be kept at arm's length.
Yeah, I put it on my CV as public scholarship, and would advocate for young scholars to do that same thing, because I think it is important, along with those papers and grants. More people, read my articles on popular science sites than read my publications.
So a quick comment. It's interesting, the general interest right now in science, I think across all ages, is my hope for the future. I think that out there today, there is a new crop of scientists who will hopefully take some of the learnings from this pandemic and bring them forward as they mature and they go through their careers. So on a positive note, I actually think that just the general public awareness and excitement around science could have a long-term impact in terms of how the field grows and shapes.
All right, we could go on for a long time. I'd love to, but our time is up. So let me thank the participants, Peter Hotez, Geraldine Miller, Tara Smith, and Susan Weiss. It's been a pleasure chatting with you today. Thank you.
Thank you.
Thank you, Vincent. Thank you, Mary.
I want to thank the panel. I think this has been a great discussion. I'm going to add, I liked ending on the topic of communication. And I have one example from some postdocs in my own lab. Some of them are from different countries, and they've been asked to actually give podcasts to the populations where they had come from. And several of them have had the comment that came back to them that they liked hearing from young scientists, that they began to feel that they could understand science, and they began to really trust what they were learning from the scientists. So I think it's another example of why communication is important, and we need to get more young people out there. I would wonder if the contract tracers may be able to help this as well, where they're helping people understand what's happening in their lives. So with that, I'd like to again thank the panelists and thank Vince very much for being a great moderator. And I'm hoping that we'll move on to other panels now. But I think we're learning about some ideas of how to be organizing science and preparing us and the world population to be able to tackle future challenges.
Mary, thank you, Vince. Thank you, everybody.
Thank you.
Bye.
Bye.
Welcome, and thank you for joining us today. I'm Carrie Ann Burnham, Professor of Pathology and Immunology at Washington University in St. Louis and Medical Director of Clinical Microbiology at Barnes-Jewish Hospital. I'm also one of the governors of the American Academy of Microbiology. I'm very excited about this panel and session, and I'm pleased to be co-chairing the panel today with Rebecca Farrell. Rebecca?
Hi, welcome. I'm Rebecca Farrell, Professor of Biology at Metropolitan State University of Denver and co-chair of ASM's Council on Microbial Sciences, COMS. The COVID-19 pandemic has been a time of rapid innovation that has also highlighted critical limitations in our public health infrastructure. Today, we're bringing together a multidisciplinary panel of experts to discuss the important topic of investing in public health infrastructure. The objectives of today's discussion include demonstrating the value of investing in public health, describing the need for a comprehensive public health network, and highlighting the return on investment in public health, as well as discussing our long-term approaches to building public health infrastructure. Carrie Ann?
Thank you for that wonderful introduction. I think it really sums up our goals today well. And I'm so pleased to introduce our moderator for this session, Dr. Esther Babati. Dr. Babati is the section head of the Clinical Microbiology Service, an attending microbiologist and professor in the departments of Laboratory Medicine and Medicine at the Memorial Sloan Kettering Cancer Center in New York City, and she is board certified by the American Board of Medical Microbiology. Her research interests include rapid diagnosis of infections in immunocompromised people and development and evaluation of the clinical utility of molecular diagnostics. Dr. Babati is a member of COMS, and as a practicing clinical laboratory director, she's certainly at the intersection of public health and clinical medicine. Dr. Babati, thank you for being here with us today and leading this panel discussion. I know I'm so looking forward to it. And for the audience members, if you have questions as we go, please feel free to put them in the Q&A, and we'll try to incorporate your questions along with the panel's questions as we go. With Esther, I'll turn it over to you.
Thank you very much, Carrie Ann. Thank you, Carrie Ann and Rebecca for the introduction. And welcome again, everyone, to the second panel of today's symposium, which is entitled Investing in Public Health Infrastructure. As our chairs have stated, the objective of this panel, among other things, is really to discuss how the current pandemic should inform our approach to research and investment in building a better public health infrastructure. It is my ultimate pleasure today to introduce 4 of the experts that will be leading this discussion. Our first expert is Dr. Susan DeLong. Dr. DeLong is an associate professor in the Department of Civil and Environmental Engineering at Colorado State University. She has 20 years of experience working in the field of microbiology, applied molecular biology, and environmental engineering. She is currently co-leading SARS-CoV-2 molecular analysis team at Colorado State University for the Colorado statewide effort to monitor viral loads in wastewater at 20 different locations across the state. Welcome, Susan. Our second expert is Dr. Chris Karp. Dr. Karp is the Director of Global Health and Discovery and Translational Sciences at the Bill and Melinda Gates Foundation. Dr. Karp is an infectious disease specialist, an internist, and an immunologist, and joined the foundation in 2012 after a 20-year academic career at Johns Hopkins University, where his research focused on defining the mechanisms underlying the regulation and dysregulation of inflammatory responses in infectious, allergic, and genetic disease. Welcome, Chris. Our third expert is Dr. Lee Riley. Dr. Riley is a professor and head of the Division of Infectious Disease and Vaccinology at the School of Public Health at the University of California, Berkeley. He's a physician who conducts molecular epidemiology and bacterial pathogenesis research focused on infectious disease of global importance and disease of urban slum settlements. prior to his academic career at Berkeley, Dr. Lee was a member of the Epidemic Intelligence Service at the Center for Disease Control and Prevention in the Enteric Disease Branch. Welcome, Lee. And we round up our panel with Dr. Fred Tanover. Dr. Tanover is an ABMM board certified clinical microbiologist who served for more than 18 years at the Center for Disease Control and Prevention as an associate director for laboratory science in the Division of Healthcare Quality Promotion and as the director of the Office of Antimicrobial Resistance. He then joined Cepheid in 2008, where he currently serves as the vice president of scientific affairs. He has been an advisor to multiple global organizations, including the World Health Organization and the Wellcome Trust. Welcome, Fred. Hi. So I'm really excited for today's discussion, and I'm looking forward to everyone's insight and opinion. We have several key questions that we'll be addressing in the next hour or so. And I'll get started with Chris. Chris, in your role as a Director of Global Health Discovery and Translational Science at the Foundation, you are really well positioned to really look at the pandemic from a larger global perspective. Can you maybe tell us what we have learned from the global response to the current pandemic that we can then take forward so that the world is better prepared for what we know is coming next?
Thank you, Esther. Delighted to be here. So, I mean, I think it's pretty obvious to everybody, the pandemic has revealed just how unprepared the world has been for such threats. And I would say on the one hand, the pace of knowledge and tool generation for COVID has been astounding, unfortunately, only outpaced by the pandemic itself. I'd also like to point out that it's shown a really bright light on fundamental problems of health inequity throughout the world, low, middle, and high income countries alike, inequities that have been further exacerbated by COVID itself. But challenges have been opportunities, or can be opportunities, and we've learned a lot. And I think one thing I'd like to say is that I think it's imperative that we take those learnings, not just for future responses to pandemics, the way we're thinking of them, but in terms of preparation and response to the residual pandemics that plague us, HIV, TB, malaria, malnutrition, et cetera. I think that will be absolutely essential. We cannot return to the status quo. Maybe some high-level thoughts across a variety of things, and maybe start, given the propitious announcement yesterday of the BioNTech Pfizer vaccine, with greater than 90% efficacy in prevention of mild, moderate disease, which is a stunning result and actually bodes well for many of the other first wave vaccines. So I think actually there are many similar roads to Rome here. So in terms of vaccines, you know, the time to getting such data is frankly stunning. And the lessons here are A, being prepared with the underlying science. So this is enabled because of previous work on MERS and SARS in terms of validating the spike antigen, development of the platform of structure-guided design, the ability now to design proteins with atomic level accuracy to stabilize the immunogen. You know, massive parallel at-risk investment across the entire value chain, from preclinical work through worldwide clinical testing, and very importantly, distributed manufacturing. Harnessing the power of accelerated worldwide collaborative networks, and of course, the critical role for public-private partnerships. This is incredibly expensive. So it's a wonderful announcement yesterday. Of course, this is really only the very first step. We need billions of doses. So capacity is a fundamental issue. Getting to 10 billion doses is a huge issue. And fundamentally, the real problem here is one of equity. This cannot be a situation where high-income countries get stocks before low-income countries. Furthermore, with the first wave vaccines, the mRNA vaccines in any case, there are fundamental issues of ultra-cold chain and production. So it's a first step. It's a wonderful first step, but it's just that. In terms of therapeutics, I mean, let's be honest, we've gotten almost nothing out of antivirals here and repurposed drugs. And if we're going to move from being reactive to proactive in terms of viral pandemics, we need to put huge amounts of effort and resources into generating libraries of antivirals that could be developed and manufactured at global scale within just a couple of months. So, you know, looked at in one way, developing agents with cross-family activity and coronavirus family activity with clear efficacy in preclinical models against known bad actors, and take them all the way through phase 1 in humans so that we're ready. The monoclonals, you know, again, stunning speed in going from identifying the pathogen to getting clinical results, but it's still way too slow. And that's always going to be held back until we're able to move past CHO cell production to alternative host production, and frankly, past that to being able to deliver through nucleic acid technologies. In terms of clinical trials, it's been, let's be clear, in terms of dealing with disease, it's been a disaster. There have been hundreds of studies, and almost every single one has been non-informative, either positive or negative. If you look at the recovery trial, which taught us about dexamethasone, there are clear lessons. So fit-for-purpose clinical design, you know, large pragmatic trials, accessible to all, with such trials being embedded in standard clinical care. But I think we're going to focus most today on surveillance, and you know, fundamentally, I think there's a clear need to internationalize the system in characterizing and tracking and responding to emerging infections. That's going to demand capacity development, not just of organizations, but of technologies. So, you know, next generation sequencing needs to be broadly available around the world, not just in high-income countries. You know, with data stored in global pathogen clouds, with effective open source bioinformatics, et cetera. And as we all know, for real public health impact in terms of diagnosis, those places that have done well have done rapid diagnosis, good contact tracing, quarantine, and frankly, wraparound support services for those that are positive to incentivize getting diagnosed. I think a real advance here will be a digital framework for diagnostics, which actually uses geolocation of use and outcomes to enable centralized data aggregation to help with planning. But I've been very verbose, I should probably.
I think you've unpacked a lot of information, and I'm going to just take the last point you mentioned about diagnostic and maybe ask Fred to comment a little bit on that, right? Because I think one of the biggest challenges we've had from a public health or even a clinical lab perspective has been the lack of diagnostic. How can we do better next time, Fred? And what have we done this time around that was really well done?
Sure, thanks, Esther. It's remarkable to me that diagnostics has become a whole household discussion. Never before has diagnostics really been front and center as part of a public health initiative. And let's be clear, public health is all about information. We need to gather the information, to analyze it, and then turn that into action, into interventions. And clearly, one of the things we saw early on was that first link, generating the laboratory data, was a huge issue. Nobody had enough tests available. And it also became clear as the pandemic moved forward that if diagnostic companies only sold to their best customers, that regions would be left out, that there would be clear gaps in our ability to generate the data that public health needed. And so in the last couple of months, I think what's been really gratifying is a sense of the common good, as was talked about earlier in the seminar, and that is through working through the Association of Public Health Laboratories, working with CDC, working with AdvaMed, which is the Advanced Medical Technologies Association. We've been able to pool our resources and have APHL and CDC help direct those to the areas that needed them most. So this sense of working together is going to be really critical to address the pandemic from a public health standpoint. And I think that's true not only just for COVID testing, But for a number of other diagnostics, we heard from ASM a couple weeks ago that laboratories surveyed had a lack of tests for sexually transmitted infections. They'd run out of tests for chlamydia and gonorrhea. And so again, working with the CDC, diagnostic manufacturers have been able to funnel those tests more equitably, especially around the US, but also globally, to identify where the hotspots are and moving those. And so this sense of working together, I think, is a great lesson learned, and one that hopefully in the next pandemic we'll be able to move to much quicker.
Yeah, that's the hope. Lee, I know you've worked in the past investigating food outbreaks. How is that different from the way that's managed, the way we have managed the pandemic now? Obviously, the pandemic is at this level of infection, but have we not learned anything from those?
Sure. I think similar approaches could be used, but as you say, the magnitude of these outbreaks are really quite distinct. And so, there are standard ways. When I was an EIS officer, I was taught how to investigate outbreaks, and there are standard ways to do this. Well, when you have a pandemic, it's a little different. And as we just talked about, obviously testing to generate data is absolutely critical. And of course, we do that in foodborne disease outbreaks. But what's also important is the ability to respond to the test results. And that's where I think we had a problem with this pandemic. We had our contact investigation program was totally non-existent when this pandemic hit. And so, we have all these tests now that are being implemented all over the place and much more efficiently done, much faster. And yet, this pandemic has continued to increase. And so what has the test done? Well, the reason this is happening is that we don't have a contact investigation. We don't have a way to respond to the test results. China and Korea, Taiwan, Japan, Singapore, they all had response teams. They all learned their lessons from the original SARS epidemic. And so they created a whole cadre of contact investigation teams. And they had the testing program. So the combination of those two things are absolutely critical in dealing with pandemics. And that's what we do in foodborne disease outbreaks. We do a case control study based on the test results, and then identify the vehicle, and then remove the vehicle. We just have not been able to do that. And that's just totally unmasked how unprepared we are in our public health system.
So that's an interesting point, right? Because I think, Chris, you mentioned that there's a lot of inequities around the world, but some countries have done a little bit better at managing the pandemic. And those were not necessarily what we consider usually, the richest countries in the world. Do you think what Lee is bringing up is part of that? They had that experience from SARS 1, and they've managed to create public health resources, not just testing, but people to
Our fundamental response here has been diagnostics for clinical diagnosis, not for public health, right? In addition to a complete lack of contact tracing in this country, we only have been using diagnostics for clinical management. And given that 40% of people infected and able to transmit never have any symptoms at all, it's fundamental.
Yeah, and I think that added another level of complexity to COVID-19, the fact that we had a symptomatic carriage. But as we're thinking about not being reactive but proactive, brings me to Susan, right? Your expertise is in you're an environmental microbiologist, and technical microbiologists, I really never think very much about wastewater, except maybe for the risk factor for other infection. So in your line of work, monitoring wastewater is a way to also figure out where the infections are. Or can you maybe tell us a little bit about that and how that could help us manage the pandemic, or even get a better sense of when the next one is coming? How do we tackle that?
Sure. Thank you, Esther. If people are talking about, topics that didn't used to be household conversations, wastewater did not used to be one of them. And now, I've been talking on the evening news at 6 about wastewater. So, I think the reason that wastewater monitoring or surveillance or wastewater epidemiology, these various terms that are used, has really taken off is because we know that people shed the virus in their fecal matter early on in the infection, before they're symptomatic and even if they're never symptomatic. So we can detect the virus in wastewater early. It becomes an early warning signal. And in fact, archived wastewater samples from various places in the world were tested after the pandemic was recognized, and it was found that the virus could be detected in wastewater before, earlier than we realized that the pandemic had reached these various locations. So this information tells us, looking forward, if we're able to be coordinated, have a national effort, and jump on wastewater surveillance as a national effort, the moment we learn that a new disease like COVID-19 has arisen anywhere in the world, we will know where it has shown up much earlier, and that gives us a lot of time, to get on it with these other, obviously the clinical testing, et cetera, and the contact tracing, and maybe engage in some more robust social distancing and shutdown practices in the regions where the virus has reached to more quickly slow the spread. So that's I guess what I'll say right now in terms of staying on this line of being proactive about responding and preparing for a pandemic.
So do you imagine then that surveillance testing of wastewater is complementary or could replace? We've been talking about mass testing of people. Would that be a replacement for that instead, or do you think that, especially asymptomatic people, how do you find those things? Yeah.
So if you use wastewater testing well, and there's various ways that it can be applied, but one of the ways that we think about applying wastewater testing is that it triggers mass testing in a targeted way. The virus is in that region, and so you roll out those resources there. We do, a lot of college campuses are using wastewater testing at the level of individual buildings. We're doing it at the Colorado State University campus, and we use that to determine where we need to do testing of individuals living in buildings so we can identify them, do the contact tracing, move them to quarantine. And, we could do that at a much larger community level if we're able to roll out the clinical testing at that level. And the other way that we're using it is for the state of Colorado, all of this data on citywide coronavirus levels in sewage is shared with our Colorado Department of Public Health. And that can be used as another line of evidence to help the epidemiologists and the public health entities to make decisions about, where we need to be with respect to social distancing status. But it's certainly not a replacement. It's absolutely a complementary tool that should be part of our public health infrastructure going forward, because it really is a useful piece of information, which is highly cost effective. We can survey an entire building for a couple $100 a sample, and then roll out resources in a strategic way.
Yeah. Just to say that this technique has found great utility in, for example, polio eradication. So we think of this by analogy with the liquid biopsy that cancer folks talk about in terms of doing molecular tests on blood to follow the course. We call it the liquid biopsy of global health.
Very nice.
The serum, the plasma sample of the world. Fred and Lee, doesn't the CDC has an environmental branch that's built around doing something like this? I'm not aware of that, but I was wondering maybe you guys could comment on that from your experience there.
Fred,
No. My impression is that maybe public health labs do have environmental sections, and I'm not sure if that's
So the CDC has, engaged in efforts in this space. My understanding is right now, the level is more a coordination of efforts that have emerged across the country, more through grassroots local coordination to make this testing available. The testing we have in Colorado was, I can talk about it more, but it was really a grassroots collaborative effort. And CDC has now, engaged in coordinating those efforts and has other ongoing efforts. But it would be good to have this kind of activity, pre-organized and ready to roll out faster.
Yeah, no, that would make sense, especially as we're talking about building networks of public health lab, but investing in that infrastructure of having each public health lab have an environmental section, and how do you connect them all, I think would be very helpful. And then working with academics, environmental microbiologists in academia, I think, to put all of that together, I think is one of the point of having this panel and this discussion today. Does Lee, do we ever do environmental testing when we look at foodborne outbreak? It seems like those would be an area where that would have happened.
So it depends. So if the initial investigation really points to some sort of vehicle, some food product or some environment, then one would start the investigation of environmental samples or food samples. But without the epidemiologic evidence, it's probably often considered to be a waste of time. I was told my supervisor at CDC when I was an AS officer that just sampling randomly the environment is really tantamount to being a sanitation worker. And not really an epidemiologist. And so we usually try to get epidemiologic evidence first and then go after the environment.
Okay. So there has to be some a priori knowledge of something happening.
Yeah. Some to target. Right.
Got it. Fred, Cepheid's diagnostic company, any interest in developing testing for environmental testing?
Well, that moves us in a very different direction, Esther. And right now we're struggling to keep up with the need for human diagnostic testing. So I think that's where we're going to focus our concerns right now. But I think Susan makes a very good point for public health, which is that she's talking about a tool of testing that really needs to be integrated more with the rest of testing that we're doing. And it's interesting, if you listen to seminars early in the course of the pandemic, they always talked about there's two types of diagnostic testing. There's the nucleic acid antibody tests, and then there's serology. And then that sort of emerged as there's nucleic acid and there's antigen, and then there's serology. But now we're in a very different sort of spot where we're talking about we need the really highly sensitive, very rapid, like 30-minute turnaround time test up front for triaging emergency department patients or people who are severely ill, where we need to know really right now and not 4 hours or 24 hours from now. And then we need the also really sensitive diagnostic tests that are for the mild and moderate people or asymptomatic people. And they're a 4-hour, 24-hour turnaround time is just fine. But then we move to screening. Screening is sort of a new beast. It's not something that clinical microbiologists are used to doing a lot. And here we can tolerate lower sensitivity if two things are met. Number one, that the turnaround time for delivering results is 24 hours or less. A 15-minute antigen test that's reported 3 days later is really pretty useless from an epidemiology standpoint. So we can compromise on the sensitivity as long as the turnaround time is reasonable and testing happens frequently. So we're evolving our sort of strategy for tracking the disease now. But as Lee said, contact tracing is so important. So we need lots of tools in the toolbox to do this. We need the type of testing that Susan is talking about to be integrated with the rest of the testing to give us the big picture. And from a public health standpoint, that all gets focused on what's the intervention, what's the best response now with the data we have. And we've been so slow both to generate the data and integrate the data that's put our public health efforts at risk.
Yeah.
If I could just comment on that briefly, that's one of the need for speed is one of the things that we quickly realized at Colorado State University and one of the, and Metro State University, who we partner with on this project, because we are trying to give the public health, the epidemiologist information that they can act on. And I think that's one of the reasons that it was important to roll out a local effort where samples could get to us quickly, we could process them quickly and get the data back quickly, which speaks to the importance of this infrastructure being decentralized, I think, just so that you can have that rapidity of response and data turnaround.
Fred, do you want to comment on the current sensitivities and specificities of rapid antigen testing and PCR testing?
I'm sorry, was that to me, Chris?
Yes.
Sure. So I think we're seeing a whole gamut of sensitivities and specificities. And, there's always what you read in the package insert and what you see in the clinic or the emergency department. And I think we're sorting out now sort of, again, these buckets of where these tests go. And as Esther can well appreciate, many clinical laboratories now have validated not just one or two, but sometimes 5 or 6 different tests because they need to have everything validated depending on what they can actually get and use at any particular time. So we're sort of sorting out where can we tolerate a lower sensitivity, where do we absolutely need very, very high sensitivity and accuracy. And in the diagnostic realm, when the patient presents at the emergency department, you can't have a 70% sensitive test. And so these are the sorts of things that we have to deal with as laboratorians, is figuring out what's the best application of these tests, because they're all not in that nice high 90% range. But that doesn't mean these tests aren't of value, especially when you move into the screening realms. But clearly, it's not one size fits all. Not every test is appropriate for every application, especially when you're balancing the diagnostic side with the screening, population, epidemiologic studies, the studies done on college campuses or in large industrial complexes.
I think that's an excellent point, right? Because clinical labs are used to diagnose, not usually do that surveillance. And I think that at this point, we're becoming sort of, those lines are getting a little bit blurred. But when we think about public health, as all of you have mentioned, we have to invest in some of these resources, because imagine if we have home testing now, where folks are testing themselves at home, but they have a positive result. There has to be a public health a component to it for keeping track of this and tracing all of these guys. Do you guys have any sense of what sort of network has to be put in place to really have that level of mass testing be really useful to contain the pandemic?
So I'll jump in and just say that there's make 2 points. Number one, there's been remarkable innovation on sample collection and processing, which I think is really good for the industry. I mean, if you had said, pre-pandemic saliva was going to be an acceptable specimen, people would have said, no, there's no way. Yet it's well accepted and it's working well in a lot of situations. But again, Esther, to your point about data collection and networks, many diagnostic companies have the ability to go into their machines and download their data, especially de-identified data, very rapidly, in fact, in real time. And we know from studies that we did pre-pandemic with some institutions on influenza, we can take that machine data and feed it back to the clinics, and they can get some information about, rates of positivity, testing patterns and things. So I think one of the things to think about is harvesting all those data directly from our diagnostic instruments and finding a way to channel those to public health to increase our surveillance activity. When you look at all the major diagnostic manufacturers, there's a huge amount of data there that could be tapped and could be fed into public health to increase our epidemiologic impact.
And that's an interesting point you bring, because data has been another piece of the pandemic, right? What the labs have had to report to the public health lab or the CDC, and had to make that actually easier to do, because that was definitely one of the challenges that many are still dealing with. Lee, I was going to just moving on a little bit from this. You have had some interest in looking at infectious disease in urban settlements. And how do you think that the pandemic has really affected people in that setting where the resources are really limited compared to, let's say, the United States? And can we learn something from that as well?
Yes, I think so. At the beginning of this pandemic, people were saying that this virus doesn't discriminate. And that's usually true, I think, in the beginning of epidemics and pandemics. But inevitably, these pandemics end up sort of establishing it themselves in these urban slum communities of the world, and they linger. And I think this COVID-19 pandemic is no exception. I think, we're already beginning to see that. Just the very nature of these slum communities really create the perfect storm for this kind of virus to really establish itself and do harm. And there's a huge disparity already that we're seeing in terms of deaths and severe complications. And then, we heard this news about the Pfizer vaccine. Well, is this vaccine going to become readily available for these types of communities? And there is an organization, there's a sort of a coalition called COVAX, which is a coalition of WHO and many countries, and I think Gavi is involved as well. It's just really trying to make sure that if a vaccine becomes available, that it's going to have equitable access. And so, yes, there is a movement taking place, but I'm afraid that that's probably not going to be enough. And this is something that we really have to deal with, because as long as the virus remains in these types of communities, and that those communities can always serve as a reservoir for spread to the communities around them. And so, if this virus becomes endemic in these types of settlements, then it's going to stay endemic in the rest of the communities. And so this is something that we really need to be dealing with right now.
Yeah. And Susan, the type of surveillance you described that you guys are doing in Colorado, would that be transferable to places like the ones Lee just described?
Yeah, I mean, anywhere where you have wastewater infrastructure and you can collect a sample, this type of technique can be rolled out. And I mean, it's really very cost effective if you think about it, because you're serving a large number of people with one test. And so, it's fractions of pennies per person. It's a matter of getting the sampling system set up, and rolling out the resources. You have to have some kind of molecular testing facility. But I think this is readily deployed. If resources are put to it, it's cost effective and readily deployed in all types of communities that at least have wastewater infrastructure. When we start to talk about places without wastewater treatment, then we have to look at different approaches.
Yeah, unfortunately, many of these communities don't have such facilities. They're the so-called shared toilets that are outdoors, just essentially just nothing but huts with a little little holes, and so they're frequently mixed with the water sources.
Right.
So it's I think you can do it, but there's no central place that you can do this kind of things.
Right. Okay. Yeah. I mean, if you're really talking about, I guess you use the word slums, unfortunately. So, yeah, if there's no wastewater infrastructure, then, you could theoretically test, but it's important to have a well homogenized sample so that you need such a small volume to do molecular tests, right? They're just very sensitive, and you need a very homogenized sample if you want to have the data represent the community. And of course, in those kinds of facilities that you're talking about, that becomes more difficult. And then I wonder about, rolling out other things like the pooled spit testing and things that is being done at universities, and whether we can start to look at, I think we need to start being more creative then about, some of the early surveillance strategies in communities where the wastewater is going to be much harder to roll out.
Yeah.
In terms of polio, this environmental sampling of wastewater in places where there are not wastewater treatment facilities is something that's happening actually across a broad swath of Africa as well as South Pakistan. So it is
oh, well, I need to learn about that then.
So there's the existing infrastructure to do that. Or is that what you're saying, Chris?
In the context of polio, but again, this is something that is bidirectional, right? The learnings from the COVID response need to be used far more broadly, and the reverse. So. But just a comment that you made, Lee, about whether this virus is going to become endemic. Does anybody on this panel think that this is not now a human endemic virus?
Well, go ahead, Susan.
Oh no, I just I don't see space for optimism in answering that question. But if somebody else does, please do.
Well, it's one of yet multiple coronaviruses that have become endemic, and probably will be living with this one for a long time as well. So, I think one of the other points to make, because I'm not sure it's widely appreciated, is that public health is typically underfunded, especially at local and state levels. And I think there's a lot of public health departments, especially in states, that would love to do what Susan is doing and incorporate this, but there's just no funding for that. And I think one of the fundamental lessons we've learned is how truly underfunded and underappreciated local and state health departments are. Nationally, we're doing a little bit better, but again, we've seen so many public health departments stretched to the limits. And that's why we haven't had a lot of contact tracing as much as we would like to. And one of the things we need to do is pay a lot more attention to the public health infrastructure. Lee probably remembers back in the early 1980s when multi-drug resistant TB ravaged New York and Los Angeles and places in Texas. And it did so because we've been chipping away at that TB infrastructure for so long, and we needed to rebuild it up. And I think once again, there has to be a response to funding and recognize the critical value of public health at all levels, national, state, and local.
Mm-hmm. Along those lines, I mean, you can see the range of people that have jumped in to respond to this pandemic, and they're not, my background is not wastewater testing for epidemiology. And so maybe that also speaks to the lack of funding for people who are dedicated to this particular activity.
And that's sort of that leads to this issue of, again, the sort of link between the laboratory people and epidemiology people. And, I think this is something that really needs to be really improved, not just here in the country, but around the world. The public laboratory people often don't talk to the epidemiologists in public health departments. And if this could be really done more explicitly, I think we can really deal with this issue. And this whole issue of this discrepancy between testing and contact investigation is an example of this. You have this very fancy laboratory infrastructure, but we don't have the ability to respond to the data generated by the laboratory. And we have all these amazing data. We were talking about this earlier, the amazing data from the hospitals based on test results, but we don't really have public health people using the information generated in these hospitals to make public health decisions or public health plans. We can, use all the resources that the data that are generated in the hospitals as part of a surveillance system. We don't do that. You have all this metadata in the hospitals, that's linked to the lab, but then somehow this type of data are not utilized by epidemiologists because they're often not easily accessible to epidemiologists. Kaiser is actually a very good example where they actually do that. The Kaiser has huge access to data, and they have epidemiology people using the data to generate very important public health information. And this could be done at the national level, it could be done at the county level, it could be done at the state level, but that's not being done.
Excellent point. Data as well. One of the things that has happened during this pandemic is that infectious disease, microbiology, epidemiologists have really been put in the public eye, right? Now everyone sort of know, I want to hope that everyone knows what a clinical microbiologist is because they get quoted on TV and in New York Times. And Lee, I know that you've had, you've been quoted in some several media as well. As part of this discussion, do we have something that we need to also create a better way for scientists to communicate with the public about public health? How do we do that? And how do we prepare ourselves so that the next pandemic, people know where to get the information, the correct information on how to deal with what's coming?
Well, I think what happened with this pandemic, unfortunately, was from very beginning, it was politicized. And so we had a lot of mixed messages coming from different sources. We had, people like Scott Atlas in the White House saying one thing, and then we had the president saying one thing, and then we had the scientists saying other things, and epidemiologists and microbiologists saying other things. And so there's a huge confusion from the very early on about the right message to give to the general public. And I don't know, how we can avoid this. There's all these things that happen now with this technology of the internet. And, I don't think I'm the best person to really communicate with the general public. There are, really well-versed people who can better communicate science to the public. Maybe we should take advantage of those people, more and more of those types of people. But, I think I learned myself in communicating with the media what we need to say and what we can't say. So I don't know, maybe others have some other ideas, but.
Anyone else has a sense of how we should communicate with the public a little bit better?
Yeah, I think there was a comment made earlier in the symposium that, we're told early on that we have to communicate at a 6th grade level to the American public. But, I think that's a very 1950s, 1960s view of the American public with the access to information they have to the internet now. I think we have to be clear, I have to be concise, and we have to be out front as scientists and as physicians to tell people what the science are saying and make it clear to them what data we have and what's the sort of the value of that. If we have a 90% efficacy in a vaccine, does that mean it's going to be 90% at the a year from now, or will all vaccines be 90%? We have to give people a sense about interpreting those data, helping to understand those data each step of the way.
Excellent point. 90% sounds really great. So I think everyone has to make sure they understand what that means. And that's for everything else, right? You were talking about sensitivity and specificity. 70%, okay. And so having a simple way to communicate that would be great. We actually have a question from the audience. Is there a way that academia can actually help train and push more scientists into public health? Maybe Susan, you want to start with that?
Well, I mean, certainly in the academic world, we train people. I'm an environmental engineer by training. I do think that there's an increasing interest in my discipline in engaging public health perspectives. And, we have rolled out like a minor in public health for environmental engineers so that you can train more people in these interdisciplinary spaces. I like some of the things that were said here and maybe in the past panel about simply the increase in interest that happens when things are in the news and in the newspaper. You know, students come to the university and they're interested in the things that they've been hearing about that have impact on society. So I think that, you know, actually the messaging that people receive in their day-to-day lives is maybe one of the strongest forces for increasing training in public health, because the students come to university telling us what they want to study. And then it's just a matter of directing people to the right programs. I think the programs exist, right? And so we can respond to that interest. And so hopefully the interest in science and public health broadly will grow because of the apparent need and potential to have an impactful career.
Yeah. Any comment, Lee?
Yes, sure. So in the past, many years ago, CDC used to have this program with academic institutions called Public Health Liaison Program. And so many of the CDC staff joined the faculty on a temporary basis in schools of public health in different parts of the country. And really this enabled the students in those academic institutions to be exposed to real practical hands-on epidemiology type of work that organizations like CDC do. And so I think that really created an opportunity for these students to really get into practical public health. You know, we don't need more people going into academic public health. We just need really people who are going out there and really, you know, doing the field work. Field epidemiology is what CDC trains people to do. And you know, having this kind of connection with public health organizations in academia can really generate those types of students or people.
That's an excellent point. The collaboration across these organizations really enables students to get the training that they're looking for. So yeah, that's fantastic.
Yeah.
And also, I mean, ASM has their American Board of Medical Microbiology certification program, which is clinical microbiology and public health. I think this is the time to strengthen the and public health part of many of those programs and to truly integrate them into the local public health departments. When I was training in Seattle, I had lots of contact with the state lab, with King County, and it really helped me understand sort of the fundamental basis of diagnostic micro across the spectrum. And I think that's something we need to bring to the forefront now, the importance of public health and being trained in those disciplines as well as the traditional diagnostic laboratory disciplines.
Yeah, definitely on clinical micro and public health. Fred and Chris, your background in public health, but now I guess you guys would be considered private sector, right? Do you think that as we talk about training, academics training for public health, we have other avenue where that background and that knowledge would actually be helpful? Can you maybe guys comment on how your job is really informed by your experience in either academia or public health, Fred, at the CDC?
Well, I'll just say quickly that once you put on a public health hat, you never take it off. So working in industry, I think a lot about public health laboratories and what are the tools that they need, as well as what a diagnostic microbiology laboratory needs. So I think that that's always in the background there. As I said, anything we can do to strengthen that, the better off we're going to be. Chris talked about public-private partnerships. So important in this day and age to get our message across.
Yeah, just to take it up one level, Fred, in terms of something you said before, fundamentally, what's obviously needed is a lot more money going in a concerted, strategic way into public health. Training people is fine, but if there's not actual funding for the work and the systems aren't there, we're not going to get anywhere. So,
yeah. So it's interesting when you think about public health and pandemic. If things are funded and work really well, no one knows you exist, right? Because there is no disaster. And trying to motivate people, now it's easy, sort of, because there's a pandemic and you know the importance. But really the goal is to make sure that this does not happen, that you stay sort of invisible. Susan, you wanted to add something?
No, I disagree.
Okay, excellent. Any additional comments as we're getting close to the end of the session? This was excellent. Thank you all for your, oh, wait, we have one more question. Let's see. All right, could you comment on the optimal relative balance between institution-specific assay development versus commercial assays, both during pandemic and outside of the pandemic? And I'll throw that one to maybe Fred to get started. We have a couple minutes to answer.
Well, sure. I think it's all hands on deck right now, especially early on, to try and provide the data we needed. The key thing is that whatever assay that you're using as a laboratory person, you need to know its specifications. You need to know how sensitive, how specific, what the predictive values are. And it may seem like a lot of work, but years ago when I was doing some training at Berkeley, they said, you have to learn to go slow to go fast. And that is, you need to get it right up front. And that's so important in public health. So yeah, I think there's a room for lots of these assays. The colleges that are doing their own testing, more power to them, as long as they understand sort of the pitfalls of what they're doing and that they make that link to reporting to public health. They're generating a lot of data, and that data needs to be shared so the appropriate interventions can be made. But again, develop the test, but really do the homework up front to know the accuracies and the limitations of those tests. That's absolutely critical, whether it's a homebrew test or a commercial diagnostic test.
Excellent. All right. And I think, that was something a lot of labs had to do, right? Especially at the beginning of the pandemic when there weren't any testing. But they also worked a lot with some of their basic science researchers to do this. And that is definitely something that moving forward, that collaboration or that national guard that Robin and Stefano sort of described in the New York Times article, we need to build those infrastructures so that next time we know how to approach this faster, hopefully, and still being accurate. Okay. Let's see. Yeah, there's a question about what the role of the WHO was in addressing the pandemic. I guess helping build, right? We've been all talking about building for the next pandemic. So definitely all this public health working with the WHO is something that's going to remain important. So I want to thank everyone again for this. This was really great. You guys had a wonderful insight, and I really appreciated you guys taking the time to have this discussion. And I'll turn it back to Carrie-Anne.
Thank you. That was just so wonderful. And on behalf of the Program Steering Committee, the Academy, and COMS, I would like to extend my sincere gratitude to everyone for your participation today. And thanks, Esther, for helping lead us through such an exciting and informative discussion, and just to all of you for sharing your insights, energy, and expertise. And, I must confess, it's not every day I listen to or participate in a panel that includes words like wastewater and saliva. So here we are. I would also like to thank the ASM staff with a special shout out to Win-Win for all of the hard work to make this event happen today. So now the meeting is going to move into a 45-minute break. Please use the same link to rejoin the meeting at 1:45 Eastern. And we look forward to seeing you then for panel 3, which is going to be focused on building reliable tools and effective scientific partnerships. Thank you again to our moderator and panelists. Thank you all for joining today, and goodbye for now.
Thank you.
Thanks.
Yeah, thank you so much.
Bye.
I'd like to welcome you all back to the ASM Virtual Symposium on Microbial Science Research in the Post-COVID Environment. So it's my pleasure to introduce panel 3, the topic of which is building reliable tools and effective partnerships. So throughout the morning and early afternoon, we heard a lot about silos and about how one of the problems that we've experienced that's really held things up is the fact that people are in these different areas and that there's been a lack of communication and understanding across these. This came across in both the the pre-meetings that we had for the panels, and also the keynote and the 2 panels we've just heard, and that these silos relate not just to the microbial sciences, but even beyond science itself. So in this panel, we're going to be talking about, what are these silos and why are they there? And really thinking about what partnerships actually need to be formed and between who, and how should we be facilitating those partnerships. And in doing so, we should be thinking about the obstacles that are preventing us from forming those partnerships and really getting to the root of the problem. So we heard a little bit about this in earlier sessions where there was some discussion about the reward systems that we're using. And also in the pre-meeting for this panel, the topic came up about how the currency for the reward system really differs across fields. So I think that's something that we should be discussing too. And also that there's a lack of understanding of areas outside our own area. So we don't really know who the people are or the topics. In addition to thinking about the partnerships, this panel is also going to think about the tools and resources that are going to be required for these partnerships and also for that work to actually get done. So what things are needed and how are we going to assemble those? Is there anything we can do to facilitate that? It is our hope that at the end of this discussion, we're going to come out with some concrete ideas that will help us to move forward in addressing these questions. But at the end of this panel, we're hoping it's not going to be the end of the conversation. Obviously, this is just getting started and people are starting to meet one another. And so it's our hope that this will continue. So as the panel is going on, I'd encourage everybody to ask questions. So you can do that in the question and answer box below. And now I'm going to introduce the moderator of the session, Dr. Evita Fuller. So it's a great pleasure to be able to introduce Dr. Fuller. She's an Associate Professor of Microbiology and Immunology in the Medical School and faculty in the STEM initiative of the African Studies Centre at the University of Michigan. She's an adjunct professor at Payne Theological Seminary and an ordained itinerant elder clergy at African Methodist Episcopal Church. She's a scientist who studies and teaches about viruses. Her lab has published studies on herpes simplex and influenza viruses, and she teaches medical, graduate, dental, and undergraduate students about human viral pathogens. Current implementation science research with the trusted messenger intervention that she developed engages networks of religious leaders in communities of Zambia, South Africa, and sites in the USA to help enable their effective engagement in disease prevention. She teaches intensive courses such as what effective clergy should know about HIV and AIDS, and Health Matters, what effective leaders should know, to divinity graduate students from across the USA. Her study abroad course, Global Impact of Microbes Fieldwork, immerses participants in global research field sites to explore effective partnerships for advancing health. And during the 2020 global emergence of this virus, she's been out front in transferring science-based insights to community through webinars, articles, emails, television, media engagements to multiple networks that include religious leaders, seminaries, university students, African American community leaders, and the general public. So as you can see, we have someone here who's really out of the silo. And at this point, it's my pleasure to pass this along to Dr. Fuller.
Thank you, Susie. Good afternoon to everyone. This is panel 3, Building Reliable Tools and Effective Relationships. And I want to thank the ASM on behalf of the panel for putting together this symposium. Let me introduce the amazing panelists for this particular discussion. First up, we have Dr. Michael Diamond, who is a professor of medicine and molecular microbiology, pathology, and immunology at Washington University School of Medicine in St. Louis. Dr. Diamond's laboratory studies the molecular pathogenesis of viruses and their interface with the host immune system, primarily for globally emerging RNA viruses, particularly the flaviviruses. These include Zika, West Nile, dengue, and other viruses. His group has identified many of the key innate and adaptive immune system functions that protect against flaviviruses. His group also does genome-wide screening to look at host factors that engage with viruses. And then most recently, his lab has studied the biology and pathogenesis, of course, of SARS coronavirus 2 to look at developing antibody and vaccine countermeasures and developing a novel mouse model to study the disease. Good afternoon, Mike. Welcome. All right.
Thank you.
Our next panelist is Dr. Nevin Krogan, who is a molecular biologist at the University of California, San Francisco, where he is the director of the interdisciplinary program Quantitative Biosciences Institute, or QBI, that's part under the School of Pharmacy. Dr. Krogan has led amazing work with the SARS coronavirus 2 through this institute, which now, through a coronavirus research group, which now includes hundreds of scientists from around the world. And certainly we want to know how he does that. His research focuses on developing and using unbiased quantitative systems approaches to study a wide variety of diseases with the goal of developing new technologies. He's also co-director of 3 mapping institutes looking at protein and gene networks in healthy and diseased cells, including cancer and other diseases. And we were talking earlier about how all of these come down to some similar molecular mechanisms. Welcome, Nevin.
Happy to be here. Thank you.
All right, our next panelist is Dr. Sherry Shuley, who is the acting medical and director and chief officer of all of us at the NIH, where she is setting vision for this group. She was formerly a team lead and senior advisor for disease prevention in the Office of Disease Prevention, where she led a systematic monitoring of NIH investments in prevention research and assessing the progress of that research. So important. She also has served as a program officer for the National Cancer Institute, and her work her published work focuses on the translation of genomic technologies into clinical and public health practice. Welcome, Sherry.
Thank you for having me.
And then last but not least, we have Dr. Tayyab Wasim, who is the Chief Scientific Officer at Stability AI. And if you, like me, did not know what that is, he's going to tell us later. But there he's involved in integrating emerging technologies into medicine research and politics. He currently leads the flagship program CAIAC, known as KAYAK, or Collective and Artificial Intelligence Against COVID, which I believe just began this July, which I hope he'll share with a little bit more about it for us later. He's a computational biologist with a PhD in immunology and a public policy fellow for the American Association of Immunologists. In other advisory roles, he has led the global COVID-19 artificial intelligence powered literature review project and includes serving as an advisor or member of the board for many organizations, including the WHO Working Group on Regulatory Considerations for AI in Healthcare. Welcome, Tayyab.
Hello.
All right, with all those introductions, now let's get into the conversation. And I'm going to send the first question to Mike. As we look at this pandemic and look at all that's happened with coronavirus, SARS coronavirus 2, Mike, what have we learned about interdisciplinary science and collaboration and partnership? What have you learned? You've been doing this for a long time with the flaviviruses. Can you just share some of the things in terms of building partnerships, lessons that have come from this for you?
Yeah, I think that, as a research scientist in the area of virology, historically, many of us operate in a linear fashion or linear path. We try to identify questions, we generate hypotheses, we perform experiments, we analyze the data, and then we come back and make adjustments to try to understand what's going on. I think what we've learned here is that there's another way to do science, and it's not a linear way. It is really a multidimensional way whereby you have a problem that you don't know much about. In this case, it was SARS-CoV-2, although certainly we had some understanding of basic coronavirology. but really trying to understand the basis for how this virus causes disease, how it spreads, who is susceptible, what is it doing to the host that allows it to replicate, that allows it to cause disease. And really this becomes an area where we required immense feedback from many different specialists, virtually at the same time. We needed information from computational biologists, not only to help us analyze big data, but also to begin to understand sequence variation, spread, mathematical modeling of what could happen, what would happen, what's likely to happen. We really needed clinician scientists to evaluate patients in real time to understand what is the disease, how does this differ from traditional respiratory diseases, what's going on that we have coagulopathy, cardiopulmonary syndromes, hyperinflammatory states. We really needed immunologists to help us really sort of understand in detail the role of particular cell types that might be contributing to what's going on. Geneticists, who might be the most susceptible and why. Structural biologists played an important role, an immensely important role early on in helping us predict what might be key parts of this virus to target. How is it getting into cells? How is it targeted by the immune system? Cell biologists, what are the pathways, in particular entry pathways that are critical? How is this virus actually generating new viruses? And we're learning every day it's not traditional in some cell types or other situations where you might expect it to be operating one way and it actually operates other ways. And so I think that one of the things that we've learned here is in order to rapidly identify new pathways for understanding a fundamentally new virus, we really need to have a community of scientists that are interchanging ideas, engaged with one another to create new hypotheses, rapidly test them, and create a picture that actually is not a linear process.
Yes, and that then brings the question of how do you do that? What critical tools? Absolutely. This has happened so fast and we've learned so much so quickly, and yet so much more to be determined. So Nevin, let me ask you, what critical tools do we have already, and what tools do we need in order to do what Mike has described and what many scientists, including you and your group, have been doing? How do we do that? What tools are needed? And how do you manage a collaboration with hundreds of scientists around the world? Can you just please share about that?
Sure. I think there's tools at multiple levels. I mean, one first tool here is just simply communicating with a scientist, not just in your own institution, but scientists around the world that are working together. And I've seen the program Slack being used. I didn't even know what Slack was before the pandemic, to be perfectly honest with you, but I've seen science move so quickly as people are on Slack and sharing information in real time in very intuitive ways. So you have communication tools, such as this. There's databases that are out there where groups can put their information, and that's good. And I think they need to be improved in order to peruse this in a more intuitive way. I would argue that where we need more tools is visualization. So, computational tools, putting information together computationally, I think that's a bottleneck, but I think an equal bottleneck is visualization. How do you put this data in a format where people can look at it and generate hypotheses about, what a given protein is doing? And, along those same lines, I think we need more tools in terms of integrating information together, integrating information from the scientists that Mike just went through. How do you get a data structural biology with systems biology, with cell biology, with somebody studying cell-cell context? The underlying data, I would argue, is actually quite similar, more similar than I think people recognize, but we still need to optimize the tool to be able to merge this information ultimately together. And other tools at other levels, getting these different scientists together, again, that Mike talked about. I think we need tools from funding agencies where they're putting money in to groups that are multidisciplinary. As we all know, certain institutes at NIH, like NIGMS, is going away from that type of science. That's not to say that there shouldn't be, focused R01 type science on individual problems. Absolutely, we need that. We absolutely need that. But I think getting away from this kind of collaborative type of effort is a mistake. And it's a mistake from one of the biggest institutes at NIH. So I think, you can think of tools from funding agencies, putting money into programs that are truly multidisciplinary, so all these scientists can come together and tackle these problems in an integrative, and as we've seen over the last 6-7 months, in a very powerful way.
So the tools that are needed are available now. As a virologist that's worked in the lab, I know that I may not know them, but they are out there. And the question is, in terms of funding, the fact that we've functioned from a being shut in or not traveling says a lot about how we need to move into a new dimension of research. And I don't know who wants to comment on that. That's kind of an off-plan question, but let's go with it. We can do so much more now because we have these connections than what we thought we would do before, as in going to conferences to talk about science. Here we are on a conference and all of us are at our home. So could somebody just share about that, your thoughts?
Maybe I could just chime in initially. I think this comes back to even more reason to fund these multidisciplinary type of projects. So I've learned more about structural biology over the last 6 months from my colleagues here than I have in the last 20 years. And I've learned about the software that's used to analyze structural information. And we've also had suggestions on how to integrate some of the tools we have that look more broadly at systems and how we can integrate it better with the structural biology world. And that wouldn't happen if we were all traveling around going to just our meetings based on our expertise. So I think it comes back to even more that we should be focused on getting scientists together that are focused on different disciplines, because this is where the big breakthroughs, where the big breakthroughs are going to be coming in the future.
And that's less expensive than flying around too.
I'd like to add another point, is that I think getting together has changed our understanding of what togetherness is, scientific togetherness. In the old days, this was exactly what Nevin outlined, is that we would be going to other institutions and spending time or going to meetings. And I think what we have realized is that we can actually accomplish quite a lot, if not more, by targeted meetings with individuals. They may be in our discipline or outside of our discipline, and not having to spend all of this time in transit or time that is less efficiently spread. I mean, there is, of course, a downside to the personal side of it, but I think that the way we see science collaboration and the way we see sort of interaction and togetherness will be fundamentally changed going forward based upon how we've had to interact during this period of time.
Let me get Susie or Wahiman, let's get you in here.
Yeah, I was just going to actually go back and just say I think we should actually expand that definition of a multidisciplinary approach, because so often you see written in grants, this is a very multidisciplinary approach because we're taking a neuro lab and we're combining it with an atherosclerosis lab or pure immunology lab. And even beyond just protein scientists versus computational biologists, I think for true, progress very quickly, we need to bring in other fields. So, with COVID, we saw COVID-19 was a set of 250,000 machine learning readable papers where we were now able to leverage 5 million data scientists who normally would never get involved into the biomedical sphere and have them involved. Same way, because every industry got disrupted, you have economists, social scientists, you have all these different fields who now want to get involved. And I think it's on us to really facilitate that. And really, when we think multidisciplinary, really reach outside of our just pure science silos.
And I also think that this sort of opportunity to have virtual meetings and reach outside of our normal areas of expertise gives an opportunity to show our trainees and our students that they need to be reaching out, and actually gives them the venue in which to do so, whereas they probably would have been following us to our meeting of specialty in any given year.
And I think it's less expensive. I think it's an efficient use of funds, even though there are some downsides. Nevin, go ahead.
Yeah, one thing to add, I completely agree. It is, you were saving money in one ways. And I agree with what's being said here in terms of science changing and how we interact is changing. And we can be more productive in many ways. But, I do think getting together in person induces a certain type of creativity that you just don't get via Zoom. So I'm looking forward to traveling, not as much, but I do miss traveling a little bit. So I do think that there is value to get together, but I think people are going to be more selective in terms of which meetings they go to in person. So things are definitely going to change, but I don't think we should completely get rid of getting together in person.
So this is not an expected question, but just thinking about multidisciplinary collaborations, particularly among different areas, it's my experience that even just the language of understanding each other or understanding not languages in different countries, but technical language that's used between the areas. When I've had collaborations between engineers and virologists, it's fascinating to hear the students sitting around. So would one of you or all of you comment on that? Or someone comment on how you communicate across multidisciplinary areas where people have different training? Engineers think differently than virologists or social scientists.
From the NIH standpoint, I'll say that this COVID pandemic really forced us to think about common data elements and how we exchange data in a way that, not just how we talk to one another, but even how we're collecting our data and how we can share data amongst each other. And so we put a lot of effort into common data elements around this pandemic and how we can get to those a little bit quicker than we normally would to help solve the problem.
I watched this in real time during the pandemic, and it was a lot of fun where we had around 250, 300 medical and scientific volunteers, whether it's residents who couldn't go into the hospital or scientists whose labs were shut down, all collaborating on this AI-powered literature review where there was one common Slack channel that had data scientists who had nothing to do with healthcare or science in general, asking really, really important questions. They were like, well, we saw a study come out that said X, Y, Z. Why would we need to go look for other studies? We've already seen 2 that said this. And why does N number matter? You started seeing those conversations on just a different ontology on what different words mean between various groups. And that's how it has to happen. You have to put them in the same room and just kind of let them fly and ask the questions and see, oh, hey, why do you guys do it like this? This is very inefficient, which they kept saying to a lot of research. They were like, for scientists, you guys sure are going pretty slow and not adopting new technologies.
And I think go ahead, Avita.
Now, I was going to say it's painful, but the payoff is really high. Nevin?
I was just going to add to those comments. I think as scientists, we should force ourselves to interact with other scientists that are doing other things and try to find the common language. And just a couple of examples. We do a lot of, say, mass spectrometry. We do a lot of mass spec on proteins. There's a whole other world doing mass spectrometry on metabolites or lipids. Those worlds don't talk to one another, and trying to force those individuals to talk will have a huge benefit. And they're very related fields. Another example, which we're trying to kind of connect on, is we deal with connectivity at the molecular level. We look at networks and we say connectivity is in one vein, but then we're working with a neurobiologist. They say connectivity, the brain connectivity, and it's completely different. But when we get down to it, the tools that we use for analysis are actually quite similar. And because they're so similar, we can really push to try to connect the molecular connectivity with, the cellular connectivity. So I think once you force yourself to talk, there's actually more similarities than you actually realize, which would facilitate, truly multidisciplinary research.
This is something Susie introduced this panel with about the need to break down silos. And so certainly what we're talking about does that. But I wonder, in terms of the training of personnel, we usually are still trained in our discipline, virology or microbiology or computational biology or physics or whatever. So how do you have any thoughts about how this might affect training in particularly the microbial sciences? This was not on our list, but I think it's a valid question that we need to consider if we're going to look at building effective relationships and partnerships. So how does this change our training process?
I mean, we're bringing big data into every single aspect of research, whether you're an immunologist, whether you're looking at proteomics, metabolomics, genomics, anything pretty much with an omics at the end of it at this point. Slowly, we're getting all of them. But, even when you're running flow cytometry, for example, you're now using AI-powered algorithms on the back end to try to analyze the data. are the scientists actually being trained to interpret where the downfalls are, or where are some of the assumptions that normal data scientists know to make, pure researchers don't necessarily know? And I think it's important to kind of cross-train with computer science and statistics.
So if I might comment, I think that the issue about trainees is an important one. And, one of the things that can be done, you can do it either by sort of didactic programs and broaden, let's say, graduate school education in biomedical sciences to include, I think, big data. And some institutions certainly have done a much better job of that than perhaps others. And the other is on an individual basis as a PI. I think that if your laboratory, whenever projects move in the direction where you have big data, network analysis, and even if it is not your area of expertise, you set up collaborations, as Nevin alluded to. But I think what you can do for your trainees is to have those collaborations be more bidirectional. In other words, not we design an experiment, get data, and send it off for processing analysis, but really have them take part in the analysis themselves such that they understand the processes, understand the questions that are being answered, and then understand how to analyze that data. And that, I think the bidirectionality allows the computational person to know what are the key things that they should be looking for. Otherwise, it's a big data set and you can do a lot with it. But if you don't know what the biologic question is you really want to answer, then it's a lost opportunity. So for me, as an individual PI, I certainly encourage anytime we have big data to have the student now via Zoom and before, in person, go over and sit down with the computational biologist, find out what they're doing, why they're doing it, how they're doing it. And this allows them to have confidence going forward in interacting with interdisciplinary ways.
Okay. So yeah, training this way is absolutely going to be the way for it. Looking at the systems that we have, though, the value system of publication and grants, I heard somebody say in an earlier panel that when the end of the year evaluation comes down, what is being looked for is grants and papers, and then grants and papers, and then grants and papers. So if we're going to do this interdisciplinary, multidisciplinary work that where there are many authors and it's not always easy to tell who contributed what, how then do we do that in the institutions that we are part of now? What has to happen going forward to prepare us to be able to do this kind of research that the times are calling for, but at the same time do them in the places that are still holding on to much older value systems? I don't know who would like to take that question and run with it.
Maybe I could start. I think the change has to come from the scientific community, and as we were discussing now and before, and with respect to the reward system, and in terms of, as you say, publications, that we're not in the days of Einstein anymore, where it's a single author publication. There's many authors that need to contribute to these studies, and yes. One should not penalize individuals for being on a multi-author paper, and that still happens in study section. I hear that still. It's like, well, what did this person contribute? And I was like, well, that's crazy talk. It's like the fact that all these people work together, they should be rewarded, not penalized. Same thing, goes for tenure. What did this person do? I know they're on a lot of papers, but it would be nice to see a paper or a couple papers where there's just fewer people. And then the awards that people get. Individuals get rewarded, which is great. Obviously, people deserve that. But wouldn't it be great for large groups to get rewarded? Wouldn't it be great for HHMI to fund, a whole department or a whole group instead of an individual? And that would encourage collaboration as well. And then finally, as we talked about before, grants. So it comes down to money. And I think more money should be put into these larger collaborative multidisciplinary grants, where collaboration is rewarded and ultimately not penalized.
Mike, what do you think? You're probably one of the most seasoned academic scholars on this call that's been in the system a long time. Any thoughts on this?
It's a challenge, I think. I think that Nevin is correct that most of our funding agencies are oriented towards individual investigator and not highly collaborative programs. That's not true across the board. There are certainly centers, program project grants that are available, But for example, program project grants are not prioritized. They're sort of whatever money is left over. And much of the NIH budget is still investigator driven as an individual investigator or perhaps groups of 2 people or 3 people maximum because of the way things are budgeted. And so we would really have to fundamentally create some new opportunities that would really highlight requirements for real interdisciplinary science. And I think what Tayeb had said earlier, how we define that, it may be even broader contexts than we normally think of, as opposed to, you know, me being a immunologist and you being a microbiologist, and that's interdisciplinary. Hardly, of course, but I mean, in a much more broad sense. And so I think that the funding agencies need to be able to encourage this at one level. And then, you know, our infrastructure is still departmentally based a lot of places. I mean, some of them are center-based, and in those areas, they're thematically organized and you can have multiple groups. But still, I think by and large, in academic science and academic medicine, we're still departmentally based. And maybe it's that even structure, that superstructure, in some ways creates some of the boundaries that we experience today. So I think there's both at the level of the institution and the level of how we fund.
And before we move on to the next topic, I just want to say, again, it's scientists and individuals who are sitting in the various meetings at the institutions that can make these decisions and make these differences. So it takes some brave leadership and some brave followership to say we need to do it a different way and let's see how to do it. Not easy, but possible. I want to send the next question to Tayeb, who is running something called Kayak since July. Can you tell us a little bit more about Kayak and how can we leverage data, big data, and good data to inform research and clinical decisions as has to have occurred with this coronavirus pandemic?
Yeah, absolutely. So Kayak stands for Collective and Artificial Intelligence Against COVID, and we're changing that last C to crises because we're finding that the principles we extract can really be used for everything from climate change to various social determinants of health. And at its core, what we saw at the beginning of the pandemic was you had a lot of grassroots movements where lots of really good groups were doing amazing work around the world, but there was duplication, triplication. You know, there were so many groups all working on the same exact contact tracing platform, making the same exact mistakes, and no one's really learning from one another. So our goal was to connect them all in a top-down approach. So we partnered with all the multilaterals, the UN, WHO, World Bank, and said, okay, can we create a decision-making platform where we can talk to all the experts and kind of find out what the known knowns are, the known unknowns are, and, you know, in theory, figure out what the unknown unknowns are, and essentially figure out who's doing what where, what works, what doesn't work, and what is the knowledge that you were able to extract. So, you know, we threw artificial intelligence in there, but 90% of what we were doing was collective intelligence. The work was out there, the answers were there, it's just they weren't reaching the broad audience or the decision makers who actually needed them. So at a very basic framework, that's the platform we've been creating. In terms of leveraging data, actually spoke a couple of weeks ago at the UN World Data Forum, where they're creating a global data access framework. And it was, how can we use this global data access framework for targeting not only infectious diseases, but also non-communicable diseases? Because those same underlying principles are at play. It's how can we create proper ontologies? How can we know that we have high quality, good quality data? You know, where do we put in useful data and show how it was cleaned so that everyone around the world can leverage their resources and move towards a common good? And, you know, we're doing that for not only COVID, but all the various sustainable development goals that the UN put out.
So is there an answer to that? So when you talk about the unknown, you want to break that down for me? What do you mean?
Unfortunately, you had to touch on that side. No, it's so the way we start our process is we start by interviewing experts in the field, and we say, you know, when you're an expert and you go on the media, you can't just say, I don't know. You give your best guess of here's what we think the consensus is right now, but we're not sure. The platform allows you as an expert to go in and say, I know we're all saying this, but we're really only 90% confident. Here are other theories that we think might actually work. And so once you start getting a couple of experts, you start seeing those decision points line up of everyone's kind of pointing at one direction of here's where we should be allocating resources. There's just not grants for it yet, or there's not enough evidence to justify doing that research yet. And it's honestly it's like this conference right here, where you have two different views of science. You have science that's published, and then you have science that's at the conferences, where we all kind of have a general consensus of here's what we know. We just haven't published it yet because we don't have enough evidence. But this is where everyone should be looking and the branch point that you should start at. And that's difficult for the world to come to a consensus on.
What I'm imagining is circles that of known, and then the unknown that overlaps in the middle is what's the unknown, and how do we now use that to inform decisions that have to be made. Sherry, a question for you. As we look at this pandemic, and certainly I'd love to hear what you think the lessons are coming from this pandemic for building relationships and partnerships, but how will we use what we're learning to respond to the next pandemic better? Can you comment on that for us?
Yes, I think one of the biggest thing is, not only breaking down silos amongst different disciplines, but also making sure we're studying diversity of people, a diversity of ethnicity and race, diversity of geography, diversity of disease states, so that we're really understanding who's being affected, why they're being affected. Is it a structural, social determinant of health kind of thing, or is it a biological reason that they're being affected? Who's having poorer outcomes? How can we determine that? So if we're not really studying diverse populations, it's going to be very hard to turn really close and turn that crank really fast on a pandemic on the whole population when we don't know very much about certain groups of the population.
Maybe I could interject a bit. I sort of thought about this. Were we prepared or were we not prepared? And I think in many respects, we were prepared. Of course, could we have been prepared better? Yes. Where were we prepared? Well, there was a fundamental knowledge of basic science and basic virology and the immune system that allowed rapid generation of countermeasures and testing on a scale that we've never seen before. Yeah. And maybe we've just heard some promising data, at least from one of the vaccines, although it is still early, if you compare it to where we have been in the past. However, where we've not been prepared, I think we understand the differences in individual interactions and responses, as Sherry alluded to, and really understanding more about human biology at its core and how it may differ from animal models or cellular models that we're actually studying, and how we need to expand that. But I think one of the things that is quite clear is we need to be, in order to be prepared for any new pandemic, whatever it is, we really need to invest in our research infrastructure at many levels, not just at the traditional level, which may be sort of basic science, but we really need to sort of build up on many other levels as well. And there's going to be discoveries that come from that are not just going to be useful for the next pandemic, but are going to help us in technological advances in all fields.
I would agree that we have been prepared in many ways, although in other ways I would say no. And one of those areas is really coordination. And if you look, for example, at the clinical trials that have been ongoing, 80% of them are hydroxychloroquine. Like, do we need that many clinical trials on hydroxychloroquine? Maybe a couple, and then other funds could be going to other potential drugs. So I think more coordination in the future to eliminate some of this redundancy. And just to touch on what Sherry said too about studying diversity, I couldn't agree more. To me, it's fascinating. Why isn't the virus spreading as fast in Africa? It seems like people aren't talking about that. Is there something about those with African descent that are making them potentially more resistant to this virus? I think we should be looking at different parts of the world when we are studying different diseases, because different groups will be responding differently, and we can learn from that and apply that to everybody.
My fear is that it's not spreading, not because it's not spreading, it's just hadn't got there yet. That is my great fear from working in Southern Africa, that it just has not hit yet in the way that it's going to. I hope you're right, but I'm really believing it's not bad. It's just access at this point. Taib?
Oh, I wanted to comment on one of the things that Sherry said about, social determinants of health as being a research area. And it's something that, I think about a lot. It's, we have all this research that goes into social determinants of health that never really goes anywhere. We keep doing this research and we publish it and it's, okay, cool, what's next? And it's, okay, we'll do more research about it. But the whole point, most people go into research because they want to make a difference. You do this social determinants of health research because you want to see, at the end of the day, the people that you're doing this research on have better health outcomes. And it's how do you translate the research you're doing so that, data scientists can understand what it is, health economists can understand what it is. How do the health economist data that comes out get translated for policymakers who have to make that final decision, for working groups on the ground? There's no integrated pipeline like you see in drug discovery where, here's my final target and here are all the different people from marketing to product development to, real world data outcomes for a final answer. But for social determinants of health, that gets lost. Everyone's kind of just doing, here's the cost effective analysis of if we fix the waterway in this area and that's it. And here's what the social scientist says, and here's what the data scientist says, and here's what the peer clinician says. There's no integrated pipeline.
So where do we get that? How we just talked about that coordination is needed. We just talked about multidisciplinary research and changes in value system. How does this coordination happen? Are there any models that you guys are aware of? Obviously, Nevin, what you're doing at your institute is a model in some ways. So what if people want to do this, and ASM as a major leading microbial science society, the one, what would be the first things we need to do, or what are some of the things that need to happen? What are some good models?
I guess a few words I'd say about what we've set up here. One point I'd make is, in my opinion, don't just snap your fingers and say, let's get hundreds of people to work together. You need to build up.
Doesn't work like that.
It doesn't work like that. Certain examples you can do that, obviously, but on the widespread collaboration, it's hard. It's very hard. And how do you do that? You build up trust, right? And so over the last several years with our institute, we've been going around the world, having symposium, bringing scientists together, not realizing in some ways, well, why are we getting all these people at the Pasteur Institute together at UCSF, but just knowing that something good will come out of that. And the effort that we put in over the last several years, I would argue we were in a perfect position to respond to this pandemic. So it's about just relationships and people and building up trust between scientists. So then when something does come along, you can be in a much better position to respond to it.
And I would argue between scientists and the participants in the research as well. That trust is very important.
I certainly see that. As a matter of fact, the intervention that we do is called Trusted Messenger, because that whole trust piece is so essential for anyone to receive what you're saying. If they don't trust you, they're not going to hear it, and they're definitely not going to do it. Mikey, you actually had talked earlier about how we keep groups from 26 groups doing the same sort of thing, and that also comes into coordination of research. Like, we might need repetition and affirmation of data from several groups, but how do we keep multiple, like 20 different groups from approaching the same question, as opposed to using resources more efficiently, such that we're looking at a whole spectrum of things that need to happen?
So, I think there has to be sort of a couple levels. One is probably a real-time communication system where scientists who are sort of in the same field can be aware of what everybody's doing and feel it's okay to exchange that information so that you can get a certain level of, reproducibility of a finding, but not, overwhelming 20 different people doing the same thing, or as Nevin alluded to, 50 people doing a similar clinical trial. Part of that might be done centrally through granting agencies who may, canvass and ask people what they're doing to try to get a sense of it. But part of it's going to also have to happen at the ground level moving up, whereby people have to be able to sort of in real time exchange information on what they're doing. Some of that happens for some very limited labs where they may post their data in real time as they're doing it on the particular experiments. And so you can see transparently what they're doing. And then you can decide as another investigator, well, should I be doing that, or are there other groups doing it? But it's not coordinated in any way. So I think we probably need at both levels, one centrally and the other a place to interchange, to say to other investigators, these are areas that we're working. And then when there is overlap, to have a place for discussion about that, to decide which lab is best suited to do what in the context of a pandemic, and what their contributions should be and how they can work together rather than independently to address some of these important questions.
That certainly goes against the system that I guess I was raised in as a scientist, where you're concerned about getting scooped as opposed to more concerned about making progress in the field.
We have to get rid of that term.
I would love to get rid of that term, but how do we do that? Yeah, and again, I guess it's up to the scientists as we train our trainees to begin to instill different values for what's necessary in these times that we are now doing science.
But Nevin pointed it out, maybe he can comment, it's not just it's the institution, how we are assessed, how we are judged, how we are promoted, how we are valued, all right now, for the most part, is based on individual accomplishment. It's not always the case, but certainly in the biomedical sciences, there's a significant amount of that. And so that's part of the problem. And that leads to the idea of, me doing it first at the expense of you, not showing you what I'm doing until such time that it's done, this kind of approach to science. And I think that's part of the problem, is the way that we actually are evaluated.
And maybe just an extension of that. Yeah, I totally agree with Mike. But looking back on my career, and I actually tell this to my trainees, it's been better for me to go and share unpublished data overall than it has been not to. You get burned, one set of, 6, 7 times, or one set of 10 times. And, the first time somebody does that and they get burned, for the rest of their career, they'll never share information again. But more often than not, somebody comes to me and says, oh, I got the same thing. Let's put it together, or let's co-publish. And then it's more beneficial for everybody. So I tell my trainees, I said, share, be as open as,
I love that,
reasonably as possible at a meeting, because overall, more good things than bad things will happen to you.
There's a question in the chat that's relevant to this. It says it is going to be an uphill battle to change the system. This is talking about the value system. How can we convince leadership that collaborative faculty are productive faculty? Corporations have figured this out years ago. There's a question, should ASM write a position paper on this? Are we responsible individually and in our societies to change the culture such that being concerned more about getting scooped than being concerned about sharing data so we can make progress becomes the old thing, and the current and the way we do things is what we're talking about, of sharing data and being glad about the multidisciplinary studies that we do that are not everybody doing the same thing. Aren't we responsible for that?
Yeah, I would say Taya said it really well at the beginning, that we all got into research to make a difference, and then we lose that, right? So if we really want to be making a difference, we can't be worried about getting scooped and having our data out there. We really need to be putting stuff out there so that we can actually turn that crank faster.
And then we want to also think about not just communication among scientists and even social scientists. So we've talked a little bit about the need for earlier panels, historians and other disciplines being involved in the issues that surround the pandemic. So then what do we do with community groups and the public? This is talking about scientists, whether they're biomedical or social scientists, communication. But then what we've seen a lot with this pandemic is that the public must know. We can do nothing if the public does not embrace what needs to happen before we can get vaccines or antivirals. And as you see in the US, that has been the issue, that the public who controls really what happens with this pandemic somehow is not getting the message. How does that get better? What's the role of scientists in this? We have things like
I think scientists play the most central role. I think, at the end of the day, that's who the public, in theory, turns to for trust in these issues. I think, I think masks are a good example of this, because for masks, for the first time, in the pandemic, the public saw the scientific process at work. doing experiments, publishing results, getting it refuted by other scientists, switching positions based on nearest evidence coming out. So we said, you don't need mass, you don't need mass, you don't need mass, you might need mass, you do need mass, no, you don't, yes, you do, no, you don't. And then really, if you look at it, the switch in both policies as well as perception happened with the meta-analysis that was published by Jeremy Howard and the rest of that group. And it had three basic things. It was comprehensive. They took all 150 odd studies that were done on mass. It was authoritative. They had all the right people at the table, and it was up to date. And having one comprehensive body of work that the public can turn to, I think really changed. It was a paradigm shift. It changed the conversation almost immediately on what our position on mass is. I think it's up to us as scientists to foster, people doing that type of work. I think a lot of times, a lot of these meta-analysis or systematic reviews that are done are sometimes frowned upon compared to the big breakthroughs that come out in, doing basic science research. But those are very important for keeping public trust and going through the process.
I do think that,
Mike, go ahead.
Yeah, I do think one of the other things that we need to learn how to better do is to convey our advances in a measured way, to show optimism, certainly, but not be overly dramatic about perhaps single-based studies where there's not validation across what's going on. Because what happens is, as a scientist, you may be excited by your data, but you may also acknowledge that it has limitations, that it needs to be reproduced, that it may be particular to your specific system that you're studying. And yet that information does not get conveyed to the public. They may pick up on one or two things, and somehow or another, we need to do a much better job of conveying the message and limitations of science at the edge, at the frontier. Because at the frontier, it still has a ways to go before it is actually vetted in not only in the field, but also in terms of whether it's actually right or not. And things happen very quickly afterwards, and they lose control out of it. And then basically what happens is as we make mistakes, we lose public confidence. And that's far worse than making advances.
I was going to say something similar. Mike said it much better than I could have ever said it, but maybe I'll just add on to it. And, realizing, I knew that, the media, I knew it was important before COVID-19, but then I realized even more how important it is. And you need to temper, as Mike is saying, your excitement. You want to be excited, but you got to get across the message that science takes a long time. It's moving incredibly fast for right now, but not fast enough for the general public. But that's a message that we need to get across properly, and the onus is on us to do it. I believe, but isn't it great that you see scientists on instead of sports stars? I'm a big sports fan. I love to see, but to see scientists front and center. To me, I think it's just fantastic. And as Mike's alluding to, if we can do a good job in terms of communicating with the public now, science can and will remain front and center as the dust settles on COVID-19, and I think that's a great thing.
We have to be comfortable doing that.
Sorry, I know some journals have done layman terms, like you have to say what your study means in layman terms. I think that's great.
That's a great idea.
Being able to explain it very, very simply and getting that point across is so important for non-scientific readers.
But even so, I think we have to be, as scientists, write a lay summary, but be very careful about it. I can't tell you how many papers that I review that scientists who are my colleagues overstate their data, way overstated, and for the purpose of perhaps getting it published or because they actually believe it. But if you actually look at the data that they provide, they can say some things, but not others. And so that has to be transmitted not only to your colleagues, but also to the lay public. And I totally agree with Nevin. The science, the pace of it is very fast, but the expectation is much greater than the reality. You still have to actually do the analysis, do the studies, reproduce it, and it just takes time.
So, folks, we're quickly growing close to the close of this panel. I want to thank you so much for this discussion, but let's try to leave with your thoughts about what can we do, one thing that can be done to move forward with this idea of building relationships and reliable partnerships as we are talking about the need now. So if you have one thing that could help break down the silos that you would encourage people in this panel to walk away with and try to help happen at their home, wherever they are, institution, I'm going to ask each of you to give us one concrete thing. Whoever wants to go first.
I would say publish raw data along with your code to run it.
I would say as a scientist, I think one should push themselves to be in an uncomfortable positions as possible, as often as possible. That means getting into other disciplines and trying to learn other people's language.
For which you're not rewarded at the moment. Okay.
Short term, but maybe long term. Hopefully long term you're rewarded.
Okay.
I would say seek out experts who are not in your field at all and discuss ideas and try to go beyond your comfort zone and trying to attack a problem and get feedback from many other people to be able to expand your horizon on a particular problem.
I have to ditto what they said, and maybe even once you start having those conversations, like Nevin said, it's all about the incentive structure. Figure out what their incentives are. For data scientists, it might not necessarily be getting publications. That might not matter to them at all. Figure out what matters to them so that you can work together more cohesively.
I thank you all for your time and your thoughts. And I'm just in my own mind thinking that we have to do what we have to do, whether it's valued or rewarded or not. And I know that may not be popular and that may not be the most self-serving thing, but I think in the long term, when you're doing what you're supposed to do and it makes a difference, you can argue that with anybody as well as you're happy with yourself seeing it make a difference. So I think we're all in different places and spaces and bring a lot to the table. Thank you so much for a very stimulating panel. I hope we have more discussions along these lines for the ASM and others. Thank you so much.
Thank you.
Thank you very much.
Thank you.
So,
so I'd like to thank the panel and also the moderator. This was actually really mind-blowing. I was taking lots of notes, and it's going to take me a while to synthesize all of this. So yeah, I really appreciate everything that you have all done, as I'm sure would all of the other attendees of the meeting. So I'm going to go directly into the closing comments, and then I'll hand this over to Arturo after that, because I think it kind of ties in with what we were talking about here. So in thinking about, the whole meeting and all three panels, for me, there were really two themes that emerged. So one of them was the communication with the public. And I think that all three panels really addressed that and discussed that as being an issue and something that we really need to address. And the other topic was about the way we behave. So this is about getting out of our silos, being more collaborative, sharing of tools and resources, looking at how we reward each other for what we do, and really this idea of really getting ourselves out of the ivory tower. And as I was thinking this through, I thought, well, perhaps there's a link between those two things, that maybe part of what we need to be doing in terms of communicating with the public is really thinking about how we behave as scientists and how we go about doing our work. Then if we start to embrace some of the ideas we've talked about today, that it might actually change the way the public views what we do, and just make us more likely to actually communicate to them what we're doing. So I just said that was my thoughts as I was listening to all of the conversations going through the three panels. I'd also like to say that obviously a lot of topics were raised, and I think this is really the beginning of a lot of conversations, and hopefully some of the collaborations and across fields are going to be enhanced through the meetings today and through these conversations. And certainly I'm planning that within comms that we're going to continue these conversations and possibly think about whether we want to have some follow-up meetings to get more into some of these topics. One of the things that I'd like to sort of get us to think about is, as we're talking about all of this multidisciplinary activity and partnerships, and through education, making each other aware of all the different areas, that we think about how we're going to balance that with the level of depth that's going to be required within each of those fields. And, you know, whether or not we need to have different people doing different things to ensure that that actually happens. And then also there was a lot of discussion about the reward system. And obviously there's going to be a lot of problems and a lot of issues there that, you know, that's going to need to be broken down into multiple parts. So anyway, with that, I'd like to thank all of the panelists for all three panels and all of the moderators. I'd also like to really acknowledge, you know, how fantastic it's been to collaborate with the Academy on this project, and I'm hoping that there's going to be many other opportunities to do so. I'd also like to thank the other steering committee members. So that was Rebecca Farrell and Mary Estes and Marianne Burnham, who you all met through the other panels. You know, they all worked really hard on this program, as did the ASM staff. I'd also like to thank Victor Rader for his support as ASM president. So with that, I will pass. Oh, sorry, I almost forgot. I also would really like to thank the staff who worked so hard on this program, and particularly Win-Win. So with that, I'll pass you over to Arturo. Thank you.
Thank you, Susie. It was a remarkable meeting, and I listened so much, so carefully to all the things that were said. And I think my take-home impression is that this calamity will change science. We're not going back to the way things were being done. The question is, some things will be better, some things will be worse. The question is, can we take the spirit that in which many of us basically refocus what we were doing and work collaboratively with many to tackle other great problems that are out there? And I think that the answer is yes. Obviously, since we can do it on COVID-19, we can do it on other things. But the question would be whether it'd be worthwhile to have a discussion. So much of today was spent on talking about ways in which science could function better. And one of the things that it's very clear to me is that the incentive system needs to be changed. I imagine a world, for example, in which promotion was based more on collaborative papers than on first or last author papers. That would totally change the economy. You'll be looking for ways to collaborate because you know the promotions committee will give those papers more weight. You know, imagine a science where credit is given only with confirmation. So you have a great result. You may then want to find a partner to reproduce it. So you already be sharing information instead of worrying about being scooped. The idea would be to find a partner lab that would share in the discovery, because both labs will then get the credit together. That is a different economic system that we have now. If you were to put changes like that in place, the entire structure of how we do business will immediately change, because people would move to their to whatever system they would need to get their grants and to get their promotions and done. So I do hope that this is the beginning of a conversation. This is the beginning of a conversation by which we can do better in science. And I think we will. I think that COVID changes everything in many ways. It won't change the need to do rigorous work. It won't change the need to do reproducible work. It won't change the need to go deeper and to take on some of the problems. But it could change the way on how we do science, and it could change the way in which we could give credit. And I think we have an opportunity here, because of this enormous jolt to the system, to begin to imagine, if you were to design a scientific enterprise from scratch, how would you do it? And with that thought, I would end. But before I end, I want to acknowledge the many people who contributed to this. I want to acknowledge Kelly Andrews, who was our meeting and technical lead for the event, Chloe Lee and Latonia Tower for the meeting and technical support, Hannah Manto for logistical support. Michelle Bogdanovich is our comms staff liaison. I want to thank the ASM communication and marketing team. I want to thank our leadership. I want to thank Victor Rita, who opened the meeting, Stefano for his leadership at ASM, Jonathan Stevens Garcia for his leadership at DAM, but I want to give the share of the credit to Win. Win thought of this, and Win has been the driving force in getting this to happen. And I think he gets not only a lot of the credit, but he should get a lot of support for the many ways in which he is pushing the academy to a better place. And with that, I will end. Thank you.